भाऊ फंक्शन चालू करतो. हो सर, करूया.
Yes. A very warm morning to everyone from India. On behalf of the organizing committee of 3-day virtual international level conference, I am Pirzada Sadiq Ali, feels privileged to welcome honorable dignitaries Dr. Apurva Prashant Hire, the coordinator of Mahatma Gandhi Vidya Mandir Institute, honorable guest, Vice Chancellor, Professor Dr. Nitin R. Karmarkar, Honorable Dr. Manohar Chaskar, Dean of Science and Technology, SPPU, Honorable Principal, Dr. B.S. Jattade, and Vice Principal, Professor Dr. N.B. Pawar, Senate Member of SSQ, and Honorable Dr. V.S. More, Joint Secretary of MGV. I would also like to welcome renowned professors, Dr. Mrunal Bhardwaj, IQAC Coordinator and Head Department of Psychology, Professor Dr. Kishore Nikam, NAT Coordinator and Head Department of English, Dr. S.S. Chobe, Academic Supervisor, Professor Dr. K.H. Kaparnis, Co-Convener of the Conference and Head Department of Chemistry, Professor Dr. T.B. Pawar, Coordinator of the Conference, and Dr. V.A. Adode, Coordinator of the Conference. I also wish to welcome industrialists, research scholars, and last but not the least, our all dear students to 3-day virtual international level conference on current research in chemistry and nanoscience, which is being organized by Loknete Venkatrao Hire College in association with Mahatma Gandhi Vidya Mandir's Board of Studies in Chemistry as a part of the Golden Jubilee celebration of the college. The college, Loknete Venkatrao Hire Arts, Science and Commerce, is established in 1971 and ensures imparting quality education with the spirit of Bahujan Hitaye, Bahujan Sukhaye. The college has been re-accredited with A grade by NAC and has also been awarded Best College by Savitribai Phule Pune University. The theme of the international level conference is Chemistry and Nanosciences Advancements and Application. The theme of the conference reminds me the words of wisdom by Albert Einstein. He said once, if we knew what it was we were doing, it would not be called research, would it? It is the conference where we get opportunities to test knowledge and understanding. We look forward to get an exposure about what the best of the brain think about the very dynamic issues in current research in chemistry and nanosciences. Now I would request Dr. K.H. Kaparnis sir to deliver concept note of the conference.
Thank you very much. Honorable Dr. Apurva Hire, Coordinator MGV Nasik, Honorable Dr. Nitin Karmarkar, VC SPPU Pune, Honorable Dr. Manohar Chaskar, Dean Science and Technology SPPU Pune, Honorable Mama Sahib More, Joint Secretary MGV, Principal Dr. B.H. Jagdale, all the dignitaries present, my dear friend Nandu Pawar sir, then all the principals who are present over here, all the supervisors who are present over here, and dear professors and researchers. On behalf of organizing committee, we warmly welcome all chemistry and nanoscience professionals to the International E-Conference entitled Current Research in Chemistry and Nanoscience, in short, CRCNS-2022. Organized by Department of Chemistry and IQAC of LVH College in association with Board of Studies in Chemistry, MGV, on the occasion of celebration of Golden Jubilee year of our college. The main aim of organizing the International Conference is to share and upgrade the knowledge of each and every individual in this fast-moving information era. We have given a nice opportunity for those who have a thirst in knowledge, thirst in knowing the present chemistry and nanotechnology advancements, and furthermore, share their thoughts. Moreover, this conference will likewise facilitate the participants to explore and share various novel ideas. The conference intends to bridge the researchers working in scholarly community and other professionals through research presentations and keynote address. In current trends of the chemistry and nanosciences, it reflects the thriving interest in the field of research and practice for contribution and better opportunities in academics and industries. You will get ample opportunities to augment your knowledge and network. I would like to add here that the Department of Chemistry runs a research center in chemistry. 3 PG programs we are running under SPPU, that is Physical Chemistry, Organic Chemistry, and Inorganic Chemistry. The Department of Chemistry faculties have been published over 250 research articles, while near 4 patents are there in the name of the faculties. The department has 4 patents to its credit. The research center has 4 research guides. Now, one more research guide is there, 5 research guides, and 3 associated research guides. Last year, since the establishment of the research center, our 4 students had been completed their PhD, and 2 more students are going to complete their PhD at the Pune University. We want to thank in advance the conference committee for extending their valuable time in organizing the program, and all the authors, reviewers, and other contributors for their sparkling efforts and their belief in excellence. We look forward to learning the most recent results from top industrial, government, and academic scientists on these topics and everything else that is presented. We are without a doubt in a period of extraordinary advancement in current research in chemistry and nanosciences. So come and join the research and report your results. I add my best wishes for a successful and fruitful conference, and my thanks to all members who have worked hard to make this conference a grand success. Thank you. Thank you very much. Over to Pirzada, sir.
Thank you very much, sir, for delivering concept note of the conference. Now, as per our tradition, to felicitate all the dignitaries and guests. So, firstly, I would like to request Honorable Dr. Apurva Hire, sir, to felicitate Honorable Vice Chancellor Professor Dr. Now, to felicitate Honorable Dr. Manohar Chaskar, I would like to invite Honorable Principal Dr. B.S. Jaktare, sir.
Thank you, sir.
Now, I would request again Principal Dr. B.S. Jaktare to felicitate Honorable Dr. Apurva Hire. I would like to request Professor Dr. K.H. Kaparnis to felicitate Principal Dr. B.S. Jaktare. Thanks for technical support team for wonderful exposure. Now I would like to request Dr. V.A. Adore to introduce our chief guest and guest of honor.
Thank you, sir. Am I audible, sir?
Yes, you are.
Good morning to one and all present here. Myself, Dr. Vishnu Adore, co-coordinator of this international conference, welcome you all to 3 days international e-conference on current research in chemistry and nanosciences. it gives me immense pleasure to introduce Chief Guest, Honorable Dr. Apurva Bhavire, Keynote Speaker, Honorable Dr. Nitin Karmarkar, and Guest of Honor, Dr. Manoj Chaskar. It's my great privilege to introduce the Chief Guest of the inaugural session, Honorable Dr. Apurva Bhavire. Honorable Dr. Apurva Bhavire is a coordinator of Mahatma Gandhi Vidya Mandir Institute and ex-MLC Maharashtra State. Honorable Dr. Apurva Bhavire has completed PhD in chemistry as well as in management. More proudly speaking, Honorable Dr. Apurva Bhavire has been continuously working towards uplifting the downtrodden, the farmers and the laborers. Under the visionary leadership of Honorable Dr. Apurva Bhavire, the MGV Institute is continuously going towards excellence. Some remarkable achievements I would like to add here. Honorable Dr. Apurva Bhavire received the prestigious the President of India, Dr. Shankar Dayal Sharma Gold Medal Award in 2001 at the University of Pune. He has been awarded with Acharya Devendra Gaurav Sanman in 2001 and 2002 for his significant contribution in the field of education. He has received Vidya Ratna Award in 2003 from International Institute of Education and Management, New Delhi. He has been awarded with Mahatma Jyotiba Phule Award in 2005 by New Delhi Bharatiya Dalit Sahitya Academy 21st National Dalit Sahitya Sammelan on 20 and 13 December 2005. On this occasion, I would like to add here that many professors have been visited countries like USA, Italy, Japan, Switzerland, Mauritius under the visionary leadership of Dr. Apurva Bhavire. With this brief introduction, I would like to thank Honorable Dr. Apurva Bhavire for accepting our invitation for the inaugural session of this conference. Now, I'm very much grateful to introduce the guest of honor and keynote speaker, Honorable Dr. Nitin Karmarkar. Professor Nitin Karmarkar has taken over as Vice Chancellor of Savitribai Phule Pune University on 18 May 2017. Since then, he has brought in number of changes in the Savitribai Phule Pune University. Some remarkable changes I would like to add here, such as implementation of choice-based credit system, strengthening of research infrastructure, sub-centers, and skill development centers. Professor Nitin R. Karmarkar is well-known scientist in the field of igneous petrology. He has successfully guided 4 PhD students, published more than 60 research papers in the Journal of International and National Repute. Sir is the recipient of Professor C. Nagana Gold Medal of the Meteorological Society of India, Mysore. The National Assessment and Accreditation Council, Bangalore, has nominated Sir as an assessor of NAC. In addition to this, Honorable Chairman, University Grant Commission had nominated Professor Karmarkar Sir as a member of the Governing Board and Governing Council of Consortium for Educational Communication for the period of 3 years. Thus, I can proudly say that Savitribai Phule Pune University is rapidly emerging as premier institute of higher education at the national and international level under his proficient and dynamic leadership. Thank you, sir, for being present here and blessing us by your gracious presence. Now, I would like to introduce guest of honor, Honorable Dr. Manoj Chaskar, sir. Indeed, it's my great honor to introduce Honorable Dr. Manoj Chaskar, sir. Sir is the Dean of Science and Technology, Savitribai Phule Pune University. Sir has research expertise in nanomaterials and their applications, especially photocatalysis. Sir has worked on many professional bodies of universities and UGC. On the occasion like this, I would like to add here that Sir is the recipient of Young Scientist Award in 1997. Sir has completed 3 major research projects as a principal investigator, which are financed by UGC and Savitribai Phule Pune University. Thank you, sir, for gracing us by your presence. With this, I would like to hand over this session to Pirzada, sir. Thank you once again.
Thank you very much, sir, for introducing guests. Now, I would request all the dignitaries to inaugurate Book of Abstract. Support team for wonderful exposure once again. Now it's time to enlighten our brain by listening to our guest of honor, and all delegates. Hence, I would like to invite Honorable Vice Chancellor, Professor Dr. Nitin R. Karmarkar, to deliver keynote speech.
Thank you and good morning to every one of you. It's a pleasure, in fact, to be with you all for the Golden Jubilee celebration of the Venkatrao Hire College, one of the premier institute located in Nashik. In fact, it's perhaps one of the best institutions which to an extent catering to the needs of all students belonging especially from the lower or weaker sections or economically backward class, I would call it like. And in Nashik, obviously, there are 6 areas which to an extent are tribal. And this particular institute been providing education right from school to the college level. I had the opportunity, in fact, to visit this particular institute, and I was delighted actually to look at basically the kind of facilities they provide and the kind of education that gets imparted as well. So keeping this in mind basically, and on the occasion of the 25th years of celebration, a 3-day workshop has been organized, especially in the field of chemistry. So the recent developments, both in chemistry and nanosciences. We all know that, in fact, during the pandemic, we have seen the importance of basically this particular branch as such, whether it is in the development of any vaccine or many other medicinal sort of products, wherein, in fact, chemistry has been playing an important role as such. And I'm happy that you would be discussing and deliberating on this very important branch as well. On the campus of the University of Pune, in fact, the chemistry and physics, these are perhaps the 2 major departments of the university as such, and to a great extent, which has laid the research, not necessarily only it has given basically light to the people, especially in at the national level, but in fact, it is an internationally acclaimed sort of a department, both of them as well. In recent time, in fact, we have created the central instrumentation facility, which to an extent been provided to all those who are interested in undertaking research. So the best of the high-end analytical facilities are now made available on the campus. And especially during the pandemic, you all know that Serum, in fact, which perhaps is one of the leading organizations which created the vaccine. But during pandemic, basically, they were facing difficulties because most of their samples generally get analyzed abroad. So when there was a complete sort of a lockdown, obviously, sending samples abroad was a difficult task for them. So they approached us. And in fact, we could be able to provide them perhaps the best of the results with precision accuracy as such. And then subsequently, all their samples to great extent been diverted to the central instrumentation facility. So this is what perhaps I would like to, in fact, share with you all who are working in the area of chemistry, that for any such analytical help, the doors are always open. The central instrumentation facility, in fact, provides you the best of the opportunity to get your samples analyzed, to get your research ongoing as such. In recent time, in fact, there are a couple of other developments that have happened on the campus of the University of Pune. And one among them is the Molecular Diagnostics Center. Molecular Diagnostics Center basically takes care of not only diagnostics of the COVID, but besides that, all those communicable diseases like swine flu, chikungunya, dengue, all those basically, we all know India is the most populous country, and these are the recurring sort of diseases that keeps on visiting time and again. So keeping or rather making a consortium of more than 40, 50 odd hospitals, we in fact are providing the diagnostics for these various different diseases as such. So this is a new thing that has come onto the campus of the University of Pune. In recent time, in fact, Pune has been given a specific title. It's called as a Pune Knowledge Cluster. And we are hosting, in fact, the activity of the Pune Knowledge Cluster on the campus of the University of Pune. And although it is called the Pune Knowledge Cluster, but it is catering to the needs of even Nashik, Nagar, all those areas which comes under the jurisdiction of the University of Pune as such. So this particular center basically provides visibility to both academia and industry as such. So we work together on various different aspects, including research part as such. And in research, obviously, during the pandemic, in fact, whether it is COVID testing, whether it is antigen kit testing, whether it is serological survey, whether it is the efficacy of the vaccine that has been produced, all such things been looked after together by the industry as well as the academic institute as such. And that is what, in fact, we are extending it further to the areas of Nashik also, because Nashik has a medical university. And in fact, for quite some time, I had the additional charge of this particular university. So we have signed an MOUs with them also. So that is another area wherein obviously you can think of taking forward your research activity as such, whether it is capacity building, whether it is vaccine development, whether it is environment, whether it is water, whether it is agriculture, whether it is healthcare, and so on and so forth as such. The other area that presently we are concentrating the most is making students as an entrepreneur. And in fact, we have created an ecosystem on the campus called the SPPA Research Park Foundation, which is a special purpose vehicle, a sectioned company. And we are focusing on 5 different verticals, which include water, healthcare, then agriculture, and 2 other important areas, basically, that is the electrical mobility and artificial intelligence as well. And we, in fact, are mentoring not necessarily only students for startups on the campus, but in fact, there are 450 odd colleges which have come on the page, and we have created innovation councils in these various different colleges as such. So wherein there are hackathons, there are competitions that we organize, and through which the best of the ideas to a great extent get nurtured and get supported also. The idea is basically to make them a successful entrepreneur. So we provide them basically advice in all different aspects. And then when the prototype is ready, in fact, to scale it further, we also bring in an industry partners. So this is what the kind of a basically ecosystem that has been created, and which is open to all the students. They are not necessarily meant only for the colleges of which are situated in Pune. In all other areas, in fact, we intend to extend these facilities, not necessarily only to the colleges to our jurisdiction, but beyond us. There are presently 40 startups, in fact, are working in different areas as such. So research is one thing, but basically for a professor, either he has to publish or perish. But now these days we are talking more about patenting and converting basically or translating the research into a measurable kind of outcome as such, which is beneficial to the society. And from that point of view, I believe the 3 days workshop, wherein I have seen, in fact, we have partnered with industry, we have partnered with some of the national institute like NCL. There are people, in fact, coming from all different parts, including Assam and so on and so forth. So it's going to be a feast to basically most of the researchers would be participating in this online sort of activity. Generally speaking, celebrating 25 years is always a dream for an institution as such, and it has to happen physically. But we have been passing through a very challenging time, a pandemic situation. There are various different protocols been actually assigned, and therefore we have a limitation. And therefore, presently, although not physically, but we are meeting through this online mode as such. And therefore, in fact, there are more there's more time to discuss and deliberate on various different issues. I think because through online, in fact, people are easily accessible, easily available access. So some of the best brains you have been able to brought in for this particular conference. So I'm pretty sure those who are participating, the young researchers, would get immensely benefited as such. So I wish, first of all, the Institute a glorious kind of a future. You already have put in good efforts for last 25 years. So I pray to the God that the Institute grows further, leaps and bounds, and in fact, create many more sort of researchers and students basically, or in developing the human resource that is required for the development of this particular country. And even for those students who are participating researchers for this particular conference, I wish you will have a sort of a wonderful sort of discussion and deliberation, and that perhaps would show you the right path, the areas you'd be able to in fact ponder at or rather choose for your further research activity. So once again, I thank the organizers for giving me a small opportunity to have a small chat with you all. Thank you so very much.
Thank you very much, sir, for enlightening us with your kind words. Now I would request Honorable Dean Dr. Manohar Chaskar to address the conference. Thank you very much, sir, for enlightening us with your kind words. I would request Honorable Dean Dr. Manohar Chaskar to address the conference.
Good morning. Professor Honorable Nitin Karmarkar sir, Honorable Apurva Hire, Honorable ex-Dean Mama More, Honorable Principal Jagdale, Vice Principal Nandu Pawar, all staff member, coordinator, convener, dear friends. I am happy to talk to you on the occasion of International E-Conference on current research in chemistry and nanosciences organized by Mahatma Gandhi Vidya Mandir, Loknetya Venkatrao Hire, Arts Commerce College, Nashik. We inaugurate this conference, though due to the pandemic, we away from each other, but we are in spirit together. The Mahatma Gandhi Vidya Mandir, LVH Arts Commerce Science College is celebrating a Golden Jubilee year under the dynamic visionary leadership of Honorable Prashant Dada Hire. My friend and dynamic principal B.S. Jagdale and his team took a great efforts to conduct such a wonderful international e-conference. I'm saying here such a bold statement because in this conference, they received more than 100 abstract and 50 research paper, which will be published in the good reputed journal. And this happened because a man gave a strong support to Principal Jagdale, and that is Dr. Apurva Hire. The approach of this conference is interdisciplinary and is aimed to bring together leading academic scientists, researchers, scholars to exchange and share their expertise and research. I'm pretty sure next 2 days will benefit to students, staff, and researchers. I know this institute very closely, all the management of this Mahatma Gandhi Vidya Mandir. So on this occasion, I guess give one suggestion to Apurva Bhau. Why not you start to think the one good research center in nanomaterials? Because as our Vice Chancellor just stated, you people are working in the weaker and the tribal area. So at least you have once a prepare a good proposal, because I know very well you have the various PhD research center at various institute, and you start the one research center, nanomaterials, because nanomaterials can be used for a wide variety of things, ranging from purification of water to wrinkle-free fabrics to curing cancer. And you people that have potential to elaborate, and whatever just Vice Chancellor stated, that we have the analysis facility inside the Pune University. So my suggestion, you can go ahead with good proposal. Whatever help you require, that help we will do on as a Savitribai Phule Pune University to bringing a good proposal, and then you can just with some industry people, you can send to government bodies, and hope so in Nashik district in future, MG we have a good research center in nanomaterials. So this is one small expectation from my side. Once again, I wish to all the organizer and give best wishes in this Golden Jubilee year. Thank you very much.
Thank you very much, sir, for advising and motivating us. Now I would request the chief guest of the conference, Honorable Dr. Apurva Hire, to deliver speech.
Good morning, everyone. Vice Chancellor of SPPU Pune University, Dr. Nitin Karmarkar, sir. The Dean of Science and Technology, Dr. Manoj Chaskar, sir. Joint Secretary of Mahatma Gandhi Vidya Mandir, Dr. V.S. More. The Principal of the College and Trustee of Mahatma Gandhi Vidya Mandir, Dr. B.H. Jagdale, sir. Vice Principal Nandu Pawar and all their team, the head of the chemistry department, Dr. Kapadnis, sir, and all their colleagues and all the participants. First of all, I congratulate Loknethe Venkatrao Hire College on their Golden Jubilee year, celebrating the 50 years. And on this occasion, they have organized one of the international conference in nanotechnology and current research in chemistry. Nanotechnology is the need of nowadays. As we have heard just now, Dr. Manoj Chaskar has told us to start a research center in all this nanomaterial. Sure, sir, we'll plan according to it, and if possible, we'll start it in this college only. As day by day, we are increasing the research centers in Loknethe Venkatrao Hire College. I think now the college has 5 research centers, and we are in planning to start new 3-4 research centers in upcoming 2-3 years. Today's conference, there are around 100 plus abstract papers have been reached, and 50 research papers are also there. So soon the college will be publishing the research journal. And one of the great achievement for the chemistry department of Loknethe Venkatrao Hire College also, and our Mahatma Gandhi Vidya Mandir's Board of Study of Chemistry has taken good efforts in organizing all this conference. On behalf of Mahatma Gandhi Vidya Mandir, I congratulate them all and wish them all the good luck. Thank you.
Thank you very much, sir, for your kind words. Now I would request President of the conference, Honorable Dr. V.S. More, Joint Secretary of MGV, to deliver presidential speech.
On behalf of Mahatma Gandhi Vidya Mandir, Nashik, and Loknethe Venkatrao Hire Mahavidyalaya Panchavati, I welcome all the dignitaries who attending the functions, and at the same time all the participants who are here to listen the great conference. First of all, I welcome Honorable Vice Chancellor of Pune University, Dr. Nitin Karmalkar sir, our friend and senior faculty, Dr. Chaskar, Dean, Dr. Apurva Vire, our coordinator of Mahatma Gandhi Vidya Mandir, and our young guide for giving us the motivation to have a good quality in education. At the same time, I will not take much more time because this is the technical session or technical conference, and I am from commerce. I am not related to chemistry, but I congratulate all the team of Loknete Venkatrao Hire Panchavati College for organizing such wonderful international conference or a discussion on current research in chemistry and nano subjects. On behalf of Mahatma Gandhi Vidya Mandir, all the resource persons who are here to join or to guide us is Ramaswamy from Mauritius, Avinash Kumbar, Vaishali Shinde Madam, Dr. Rode, Chief Scientist, NCL, Dr. Mahesh Patil, Moreshwar Chaudhary, and Praveen Ingode. I again congratulate all the team of Principal B.S. Jagdale and best wishes for celebrating the Jubilee year of late Loknete Venkatrao Hire Mahavidyalaya, Nashik. Thank you, sir.
Thank you very much, sir, for delivering presidential speech. Now I would request Professor T.B. Pawar, sir, to propose vote of thanks for inaugural session.
Thank you, Pirzada sir. May I yes, may I audible?
Yes, you are audible.
Thank you. Good afternoon, one and all, sir. I, Dr. T.B. Pawar, coordinator of the International Conference CRCNS-2022. I feel honored and privileged to get the opportunity to propose a vote of thanks on this special occasion of the inaugural function. I, on behalf of our college, principal and the organizing committee, to Honorable Dr. Apurva Hire, former member of Legislative Council, and coordinator of Mahatma Gandhi Vidya Mandir, Nashik, who always constantly motivate, supports, and encourage for quality and student-centric education. I also thankful to our Joint Secretary, Honorable Dr. V.S. Mori, sir. I also, on behalf of our college, principal and MGV Institute, convey deep regards and hearty thanks to our Honorable Vice Chancellor, SPPU Pune, Professor Dr. Nitin Karmalkar. I would like to thank the guest of honor, Dean Faculty of Science and Technology, SPPU Pune, Dr. Manoj Chaskar, for this kind support. I also would like to thank Honorable Professor P. Ramasamy, University of Mauritius, who accept our invitation and quick reply for speaker. I also thank all resource persons, Professor Dr. Avinash Kumbar, Department of Chemistry, SPPU Pune, Dr. Moreshwar Chaudhary, Marie Curie Postdoctoral Fellow, Spain, Dr. Mahesh Kumar Prakash Patil, PKN University, Busan, Republic of Korea, Dr. Vaishali Shinde, Department of Chemistry, SPPU Pune, Dr. C.V. Rode, Chief Scientist, NCL Pune, Dr. Praveen Ingole, CSI, NEIST, Assam, for their positive response. I would like to thank Honorable Principal Dr. B.S. Jagdale for this. The words are not enough to thank their constant guidance and support to shape the conference. I also would like to thank Vice Principal and Senate Member of SPPU Pune, Dr. M.B. Pawar, IQAC Coordinator, Professor Dr. Mral Bhardwaj, Dr. Vinit Rakives, Supervisor and HR and Finance, Dr. S.S. Chobe, Supervisor Academic and Examination, and Dr. H.D. Patil. I would like to thank Head of the Department, Professor Dr. K.H. Kapandnis, for his constant help and guidance. I am very much thankful to Vishnu Adore, co-coordinator of this conference, and all our faculty colleagues and non-teaching staff members who always stand by us and motivate us. I feel proud and thank you for making this event successful one. I would like to thank all the honorable delegates and the participants who blessed us with their presence. I am also very much thankful to all program advisory committee members and organizing committee members. If time is money, then today you have spent millions for us. Thanks to all of you for making this event successful. Enjoy the conference. Thank you once again, and over to Peter.
Thank you very much, sir, for proposing votes of thanks. Now I would like to hand over the responsibility to Dr. Vishnu A. Adode, sir, for next session. Thank you very much.
Thank you, sir. So all dear delegates and participants, so the first technical session of this international conference is going to start within few minutes. Before that, I would like to tell here that this is a 3 days international conference. On the first day, we are going to have 3 technical sessions. In the first technical session, Professor Ramasamy from University of Mauritius is going to deliver a talk on computational chemistry. After that, after the technical session first, we are going to have a renowned professor from Savitribai Phule Pune University, and he is Professor Dr. Avinash Kumbar sir, and he is going to deliver technical session 2. And in the technical session 3, Dr. Morishwar Chaudhary, who is Marie Curie postdoctoral fellow at ICIQ Spain, he is going to interact with us on polymer supported catalysis. So this is the schedule of first day. In the second day of this international conference, that is on tomorrow, we are going to have 2 technical sessions. In the first technical session of tomorrow's first technical session, Dr. Mahesh Kumar Prakash Patil from the Pokyong National University, Busan, South Korea, is going to talk on applications of nanomaterials. After that, in the second session of tomorrow's second session, Professor Vaishali Shinde from Savitribai Phule Pune University is again going to talk on nanomaterial and their application. After these 2 sessions, tomorrow we are going to have paper and poster presentation, so that we are for this paper and poster presentation, we are going to display a schedule, and according to this schedule, the paper and poster presentation will be carried out tomorrow after 2 sessions. Then on the third day of the conference, we are again going to have 2 eminent resource persons, and they are going to interact with all of us. In the first session of third day, Dr. C.V. Rode, Chief Scientist from National Chemical Laboratory, Pune, is going to deliver a talk on that current topics in the chemistry. And in the technical session 2, Dr. Praveen Ingode, who is scientist at CSIR, Jorat, Assam, is going to talk on the latest advances and coupling the chemistry applications towards the industry. That is the topic of interest from the Dr. Praveen Ingode sir. And after that, on the third day, after finishing these 2 sessions, we are again going to have paper and poster presentation, and for that again we are going to display a schedule for the participants, and after that we are going to have validatory function of this international e-conference. More than that, I would like to add here that we are going to publish the research papers which we have received from the participants, and all these papers will be communicated to the UGC care listed journal, And this UGC Care Listed Journal, the editorial board of UGC Care Listed Journal is going to review all these papers, and accordingly we are going to communicate with the participants about the decision of the manuscripts which they have sent to us. And after this process, the decision will be made on the research papers which are submitted by the different researchers, and a proceeding of the international conference in the form of one special issue will be published in the journal that is listed in the UGC Care List. So this is the overall schedule of this international conference, and any update regarding this international conference will be communicated via WhatsApp group or email. So dear participants, we are going to have the first session, today's first session from the respected Professor Ramasamy sir. And as I discussed with you, he is going to talk on that computational chemistry. Let us have, yes sir.
Ramaswamy sir is there?
He will be there within a moment, sir.
Okay. Till then, our one very eminent resource person, Dr. Mahesh Kumar Patil is there.
Yes.
So we would like to hear from him something about this inaugural function. Sir, are you listening me? Mahesh Kumar Patil, sir? This is my humble.
Yes, sir, I am listening to you.
Yes, it's my humble request to you to talk about the conference in the inaugural session.
First of all, thank you for inviting me, and it's my pleasure to deliver talk on this innovative conference. I saw nowadays it's difficult to have a visual conference, so it's really nice to attending such conference, and I am happy to present my research work in LVH College for arranging such a nice conference. Because of this, I know the students, particularly undergraduates and graduates, they are getting more ideas, and they will be happy, and they will have a path to decide their future, whether to go for PhD or research, or how to create research ideas. So they will get some more knowledge from us, the student from the LVH College. My research work with them. Usually they are working on the nanomaterials, and in my department we are working on the cancer related things. So we always used to have a collaboration. Might be Professor Jagdale sir don't know me, but I know him. I heard from Professor Vishnu Adode. He's a kind person. I got this opportunity because of him. So I would like to thank. And I think so. My presentation is tomorrow, so I will talk more about presentation and the scope of science in the particularly nanotechnology, and how it will change the students' mind to have interest in that particular area. Thank you for this opportunity, sir. Thank you.
Thank you, sir. Thank you for guiding us in the inaugural session. As you rightly mentioned, that our principal is very kind, and he gives opportunities to the downtrodden as well as the person right at the right spot. And he might be given you the opportunity, if at all, that you must deserve it. And because of that only he had been given you the opportunity. Dr. Vishnu Adode, are you there?
Yes. Yes, sir.
Now proceed, please.
Okay. Thank you, Mahesh Patil sir, for giving us your valuable time for this international conference. And definitely, we are going to have a wonderful session tomorrow. Thank you, sir. Okay. So, dear participant, So we are going to have a first session of Professor Ramasamy. So I would like to add some information of Professor Ramasamy here. Sir is basically scientist from computational chemistry group, and he is working at the Department of Chemistry, Faculty of Science, University of Mauritius. And talking about the educational background of respected Ramasamy sir, sir has received the PhD in physical chemistry from the University of Delhi. And his thesis entitled as Volume and Compressibility Studies of some amino carboxylic acids and peptide in water and aqueous solutions of sodium sulfate at 288.15, 298.15 and 308.15 Kelvin. Sir is MSc from University of Delhi itself and also BSc from University of Delhi. Talking about the professional career, Sir is the visiting professor at the University of Johannesburg. Then at the University of South Africa, visiting professor at King South University. He is professor, personal chair in computational chemistry, associate professor in chemistry at University of Professor in the year of 2006 to April 2012. And one of the important aspect I would like to add here, sir has received Best Mauritian Scientist Award in the 2016. Then he has also received the award from the Scientific Board Advisory. Nearly 60 research projects at the undergraduate level has been completed under his able guidance. 10 postgraduates, more than 10 postgraduate projects
Voice is, you're not audible. Something happened to the voice.
Dr. Ramasamy sir is not joining.
Is he joined?
Yet not, sir.
No. Okay. Adore sir, you are not audible.
Okay. Sorry for the technical inconvenience.
Yes, proceed, please.
Okay. So talking about the research publications, sir has published more than 245 research papers in the international repute. Then there are so many research projects which are funded by University of Mauritius, then the Tertiary Education Commission, then Ajman University, and all these projects have been completed under the supervision of Professor Ramasamy sir. One of the book which he has edited is Bioresources Towards Drug Development, and it is also available in the form of CD. Then Chemistry Education in the ICT Age is one of the one of the impactful book which is published by him. Then the book entitled Chemistry for Sustainable Development, Chemistry the Key to Our Sustainable Future, Crystallizing Ideas, the Role of Chemistry, Emerging Trends in Chemical Sciences and the Computational Sciences. More proudly speaking, sir is the editor of journal Open Chemistry, which is published by the D. Grittier publisher. And one of the important aspect which I would like to add here, that sir is having collaboration with many scientists across the world, and our research center, that is Research Center in Chemistry at LVH College, is also having collaboration with respected Professor Ramasamy sir. So this is the brief introduction of the Professor Ramasamy sir, who is going to deliver a talk in the technical session first. So I think sir will be there in a moment. So we are going to start the session within a moment.
Apparently, sir, we can make a small change now. Instead of Ramaswamy, I think Ramaswamy sir is not joined it. We can start with Dr. Mahesh Kumar Patil.
Sir, I have been talked with I talked with Dr. Jimmy Paulson, he has been communicating, and what reply is there is that he is going to join at track time.
Okay.
So our function has been over a little bit early. He'll be there within 5 minutes or so.
Dr. Patil is there now.
Yes, sir.
Dr. Mahesh Kumar Patil sir is there.
Dr. Mahesh Kumar Patil is there, no problem. Then let me ask Dr. Jimmy Paulson. Jimmy Paulson. Mahesh Kumar Patil.
Yes, sir. I am going to correspond to Professor Ramaswamy. Please wait. We are waiting to 12:30, and then we will take a decision. We will change. Okay, sir. I am continuously corresponding to Ramaswamy. Please wait, sir, up to 12:30, and then we will decide to.
Dr. Mahesh Kumar Patil, we are so so nice of you. You were there, but as per schedule, Dr. Ramaswamy should be there. If he is not going to join, we definitely like to continue with you, sir. So please wait.
Hello. Hello. Advisor is there?
Yes, sir, you are on.
Till Ramaswamy, we are not we are unable to connect him, and he is not joined to this webinar. So, at present situation, we can start with Mahesh Kumar Patil, or we can request to the Dr. Mahesh Kumar Patil, because he is joined today. Okay, can we change this little change in the schedule?
Yes, sir. Yes, sir. Sure.
Okay, please.
Sir,
Yes, yes, sir. I will proceed.
we will start with Dr. Mahesh Kumar Patil, sir.
Yes, sir. Yes, sir.
Please ask him, is he ready or not?
It's okay, sir. I'm ready.
Yes, sir. Yes.
So it's
I will start, sir.
Side. Will you please start? Sir, start the proceedings.
Yes, sir. Yes, sir. Okay. Dear participants, so we are going to have a little change in the schedule. Here now we are going to have a technical session first of respected Dr. Mahesh Kumar Patil, sir. For this session, I would like to request Principal Professor Dr. Arun Patil, sir, who is Principal at MGV's Art, Science and Commerce College, Manmad, and also Dean of Science and Technology, Mahatma Gandhi Vidya Mandir, Nasik. Sir, so I would like to request you to chair this session.
Yes, sir. Yes, sir.
Okay. Now I would like to request Dr. Santosh Chobe, sir, to introduce the resource person and the chairperson of this session. It's over to you, sir.
Advisor, please continue. Some technical problem is there.
Okay, no problem, sir. So I would like to introduce the resource person, Dr. Mahesh Kumar Prakash Patil. So, sir is working as a professor at the Pokyang National University, Busan, Republic of Korea. He is a highly motivated self-starter and the meritorious researcher with the proven ability to handle multiple projects simultaneously. He has more than 30 research publications in the Journal of High Repute. The sir is having good ability to work with international and environmental teams of researchers. The PhD thesis of Dr. Mahesh Kumar Patil is having a title of Antibacterial and Anti-Cancer Activities of Silver and Gold Nanoparticles Synthesized using Sosa Bareilles and Artemisia pilosa. He has worked under the able guideship of Professor Kundong Kim, who is working at the Department of Microbiology, College of Natural Sciences, Pokyang National University, Busan, South Korea. Sir is received research professor grant for 2 years from the September 2021 to August 2023 from the Pokyong National University, Busan, Republic of Korea. Postdoctoral fellowship from August 2021 to December 2023 for working under research grant received from National Research Foundation of Korea, NRF, Ministry of Ocean and Fisheries, South Korea. Young Researcher Grant for the postdoctoral fellowship for 3 years from June 2018 to May 2021. research and education from Korea Science Academy, South Korea for 10 months, March to December 2018. Okyang National University, Busan, South Korea, postdoctoral fellowship for 1 year from September 2017 to August 2018. So sir is having very good record at the national and international level and good number of publications. With this, I would like to invite you, sir, here for the inaugural talk. Now, the chairperson for this session is Professor Dr. Arun Patil, sir. Sir is basically working in the field of nanotechnology and having good number of publication in the high-class journals such as Elsewhere, Springer, etc. Number of students are working under the able supervision of Professor Dr. Arun Patil, sir. Sir is the principal at MGV's Art, Science and Commerce College, Manmad, and Dean of Science and Technology, Mahatma Gandhi Vidya Mandir, Nashik. Sir, now the session is handed by the Professor Dr. Arun Patil, sir. It's over to you. So you can request the resource person to start his proceeding. Over to you, Patil, sir.
Thank you, Dr. Arun. Am I audible?
Yes, sir.
You are.
First of all, I am thankful to the principal of LVH College and the trustee of MGVs, Dr. P.S. Jagdale, sir, that he has given me the opportunity to chair the session, because it is a very good opportunity that a young scientist, Mahesh Kumar, is going to speak on the related topic, and I have to heard it. I have to learn from the young scientist. So I am thankful to the organizing committee, the coordinator of this conference, Dr. T.B. Pawar, sir, and all the organizing committee members, that they have considered me as a right person, as a proper person rather, for chairing this session. Thank you. I am requesting Dr. Mahesh Kumar. Dr. Mahesh Kumar, I have visited your city, Busan, in 2014. I had participated in the conference organized by Maritime University. Professor Kim was the chairperson of that, and I was there for 5 days. It's a very nice place where you are working in Busan.
Thank you, sir. First of all, thanks to LVH College for organizing this conference and inviting me as a resource person. I really love to have deliver my research work. Okay, I'm going to share my screen. Okay, is everybody can see the screen?
Yes, sir.
Okay, sir.
Yes.
Okay. Just a moment, please. Good afternoon, dear all attendee and conference committee members and respected professors. Myself, Mahesh Kumar Patil, already Dr. Vishnu Adole introduced me and about my research work. Right now, I am working as a research professor in the university, Pukyong National University, South Korea. Dr. Adole already introduced about my academic careers and my research careers. So basically, I have completed my bachelor's from the North Maharashtra University, Jalgaon, and master's also. And then I worked for the pharma industry for 6.5 years. After that, I went for the PhD in the same university, Pukyong National University. After that, I did 3 years postdoc fellowship in the same university, and the same university now I'm working as a research professor. And during this time, I received a number of grants. So I have listed few grants here. And interesting, like I received a huge grant for basic research. And today I am going to deliver a seminar on the metallic nanoparticles, green synthesis, and their biomedical applications. As we know that the nanotechnology is the emerging branch, and nowadays it attracts a lot of researchers and many breakthrough science coming from this field. So I can say nanotechnology is really the ray of hope for the outcome from this pandemic issue also. Of course, there are some answers we are received from nanotechnology regarding the coronaviruses. If you read articles every day, we are getting updates and updates, and because of little contribution of nanotechnology in that. Okay, so today I will have a presentation on nanotechnology related things. So basically, first question comes like, why green synthesis? That means the synthesis comes under the eco-friendly way. And then what are the methods except green synthesis? What are the other methods? Then their mechanisms, and their what are the sources we can use for this process, like a plant, microorganisms, and how these nanoparticles forms. Basically the undergraduates and graduate students, they have just theoretical knowledge, but they never have a discussion like these things. So I think they can get better information from these presentations. And these nanoparticles have the wide applications. So I would like to explain about antimicrobial and anti-cancer because of my good research on this area only. And then I will explain about little bit about the nanoparticles of silver nanoparticles. Okay. So before going to have a research, I would like to explain about the general terminologies and general things, so the students can easily get concepts and they can understand behind my research what exactly they are listening or reading. So first thing is the terminology, nanotechnology. As we know, the nanotechnology, the term itself explains nano and technology. The nano means the dwarf, the word coming from the Greek. It means a very small or tiny particles which having the dimension about 1 to 100 nanometers. And when the nanotechnology joins with the biological things, then we can say the nanotechnology perform with the help of biological things, that is the nanobiotechnology. By using the biological agent, we can make the small nano things, that is nanotechnology. And this is interdisciplinary subject. In this subject, we cannot say only chemistry is involved, only biology is involved. It includes the chemistry, biology, physics, material science, medicine, and so many. So basically, if we see the length of nanomaterials, it's about 1 to 100 nanometers only. So whatever the things we can see naked eyes, that is different. And this is the like bacteria, animal cells, and plant cells. They are, we can see by the light microscope. The microscope we have in the laboratory, that is monoclonal or dual. And what are the things we can not see by the naked eyes or with the help of light microscope? That is the nano things, and they can be visible by the electron microscope only. Okay, so the nanoparticles, as we know, the nanoparticle, everyone is talking about the nanoparticle, but exactly don't know like what are the nanoparticles. Okay, now we know the nanoparticles is 1 to 100 nanometer particle, that a material is the nanoparticle, but they have different shapes like spherical, cubical, hexagonal. You can see on the screen like there are so many nanoparticles having different shapes. And especially the biomedical related applications, the particles like silver, gold, platinum, cerium, iron, these are like graphene, these are like more attractive nanoparticles in the field of medical sciences. Okay, like Metal nanoparticles are the nanoparticle. They can be used for the drug delivery, for the chemical reaction, accelerate the chemical reactions, or for the delivery agent. And in the biological methods, we can produce nanoparticles by using different biological agents like plant and microorganisms. At the same time, we can produce by the different methods like physical or chemical. But biological method has more advantage over these two methods because of their cost effective and eco-friendly. They cannot produce hazardous byproducts. So these green synthesized metallic nanoparticles are mostly the biocompatible. That means they are less toxic than the materials produced by the chemical and physical metals. So in this current era, like most of the papers, if you see, they are more concentrating on the activity like antimicrobial and anti-cancer things. Okay, so metallic nanoparticle synthesis can perform by basically having two approach. One is top down approach and second one is bottom up approach. Top down approach means like from big particle to the small one. Okay, like a big stone, they are breaking and making small stones. Like the same way, from the bulk part of the metals are reduced to the small parts, and this can be possible with the help of some methods I have reported here, like mechanical ball milling, thermal ablation, chemical etching, sputtering, like so many methods, but I have reported only few here. And the another approach is bottom up approach. In the bottom up approach, you can say build up nanoparticle from the small entities. For example, if you are using the metal salt, for example, silver nitrate you are using for making the silver nanoparticles, or chlororic acid you are using for making the gold nanoparticles. Same way. So this is like from small entities to make a nanoparticles. In this, you can see the biological method and chemical method. Both can be involved, even the physical method also. So here are some methods like electrochemical precipitation, soldier, laser methods, and molecular condensation methods. But these methods having toxic effect, this method, the nanoparticles synthesized by these methods having the toxic effect on the human cell lines, as per the experience reported by the previous studies. And while using biological agent, or we can say green synthesis or by eco-friendly synthesis, those nanoparticles synthesized are having less toxic effect, or we can say non-toxic. Everything is toxic at after certain limit. So we cannot say non-toxic, but it has less toxicity than the nanoparticle synthesized by the chemical or physical. So basically, if we see there are three methods: chemical, physical, and biological method. In case of chemical methods, the chemical agent mix with the reducing agent, and then we can produce the nanoparticles, and that produce or synthesized nanoparticle needs the coating to prevent the making aggregation or clusters or formation of big particles. So for that, like two steps. One step is formation of ions and then coating, and then we can say nanoparticle synthesis, and they may have like different shapes and sizes. While in case of physical methods using high level of heat or pressure or radiations to make the nanoparticles, and that may be having the different shapes. While in case of the biological synthesis, the biological substances, for example, plant, bacteria, fungus, algae, they can be used as a reducing agent, and we can make in one step by adding the chemical agent and the plant extract, and we can have a reaction in a one step, and we can get the nanoparticles, and that may varies with the different shapes and sizes. So this is like a simple three methods of synthesis. First, here we see the approaches like top down approach and bottom up approach. So the bottom up approach I am going to explain with the help of biological method. So okay, nanoparticle synthesis, as we know, the nanoparticles are synthesized, but exactly how and what exact happens. So this is just a general mechanism on the formation of nanoparticles. For example, the metal ions, maybe you can say silver ions or gold ions, when they are reacting with the phytochemicals, the plant extract. The plant extract have reducing agents. Some agents, they are able to reduce the ions into the formation of nuclei, and this nuclei comes together and they grow and they make a particular shape. Here you can say this is a circular shape. So these nanoparticles come together and make a one shape, and because of these phytochemicals, they are covered and they can make the stability to these nanoparticles. In case of chemical method, we see like synthesis is different part and coating is different part. But in case of the biological method, we have all together because of phytochemicals. They are acting as a reducing agent as well as the coating agent. Okay, so in the green synthesis, the green synthesis of nanoparticle using the plant extract. This is general flow diagram for the synthesis of nanoparticle. You can see the leaf, galls, the stem, bark, different parts. As we can extract these things into by using the just simple water, boiling with the water, and we can extract this phyto extract and then react with the metal salt. And by varying different temperature, pH, concentrations of metal or plant extract, and with the help of incubation time, we can get different shape sizes of nanoparticles. It may be silver, gold, or others. So here, for example, I have given certain characterization images. In case of the transmission electron microscope, we can see by transmission electron microscope, we can check the morphology of nanomaterials, whether it is spherical, triangular, hexagonal, or something like that, and even the shape of the nanoparticle we can see. Nowadays, there are so many softwares available online. For example, ImageJ software. If you take this image, and by comparing with that ImageJ software, you can check the size of nanoparticles. And the first step is after synthesis. This first step is UV visible spectrophotometry. This is the first step where we can say the nanoparticles are synthesized or not. There are a specific peak band where we can say this is a gold nanoparticles or this is a oxide nanoparticles. Then by using the XRD, then we can know about the crystalline nature of the nanoparticles, whether it is pure form and what exact the compositions. And by using the particular size analysis, we can analyze the average particle size. And another technique is like EDX, energy dispersive X-ray spectroscopy. We can know about the elemental composition of the nanomaterials. And by using FTIR, Fourier transform infrared spectroscopy, we can check which kind of the biomolecules from the plant material is involved in the reduction process of the nanomaterials. And after this, like the synthesized nanomaterials can be useful in the different field. Maybe it can have a microbial application in the parasitology, pharmaceutical, maybe food technology, chemical, or in the cancer things. Okay, this is about the plant-mediated synthesis of nanoparticles. And in case of the nanoparticle synthesis using the microorganisms.
Dr. Mahesh Patil, excuse me, sir. One minute, please.
Yes, sir.
1 minute. Right. Dr. Ramasamy is joined here, so I would like to first welcome Professor Dr. Ramasamy with this college, and I request, sir, there is a little change in schedule because of Ramasamy. It's unavailable, maybe because of international time slots. So I request to Ramasamy. Ramasamy, are you audible?
Yeah, thank you. I'm very sorry for disturbing you, Mr. Dr. Patil, and interrupting your talk. So I failed to understand why I was not able to join. But please go ahead, and I will carry on with my presentation. It is no problem at all. So please, Dr. Patil, you go on.
Yes, sir. Yes, sir. So tomorrow your session will be at 11:30 a.m. Indian timing. So please, sir.
Perfect. Yeah. Dr. Patil, please go on. I'm sorry again to
Thank you.
disturb this thing.
No,
Yeah.
it's okay.
Thank you, Patil
Okay,
sir.
sir. It's okay. Thank you.
Patil sir,
Yeah.
go.
Okay. Thank you, sir. Okay. Sorry for small interruption. We are now continuing with the previous section. Then here we saw like the synthesis of nanoparticle using the plant material with the different ways, and now it's time to listen about the synthesis of nanoparticle by using the microorganisms. As we know, like the microorganisms already, it's a microscopic, and you will have a question like how these organisms can be involved in the synthesis of nanoparticles. Okay, the compared to the nanomaterial organisms having bigger size, the nanomaterial is in the nanometer sizes, and bacteria having the in the micrometer sizes, that is almost 100 times bigger than that particles. Okay, so by using microorganisms, nanoparticle can be synthesized intracellularly or extracellular. Intracellularly it means inside the cell wall, inside the bacteria can be synthesized, and extracellular means outside the bacteria can be synthesized. So in case of intracellular synthesis, when the bacteria mix with the metal salt solution, that can be synthesized, the metal salt transported inside the cells via the different metal transporters present on the cell membrane, and inside the cell with the help of enzymes, that reduction process is happen and the nanoparticles are synthesized. So basically in case of intracellular synthesis, the microorganisms produce or synthesize nanoparticle inside the cell. So taking it off, we need to burst or kill the cells, and then we can isolate the nanoparticles. So it is like inconvenient process, or it add extra cost to purify the nanoparticles. So that's why the intracellular is not much explored technique, or not preferable that much, while the extracellular biosynthesis is more favorable and reported highly because of the microorganism produces something, some kind of extra cellular proteins or enzymes, and that act as a reducing agent, and they synthesize the nanoparticle. So in this case, we need to wash the bacteria biomass, then centrifuge, and then the lyse, and their cell extract can be used as a reducing agent, or we can add a complete cell supernatant into the metal salt, and we can produce the nanoparticles. Okay, for this small mechanism we can see here. Okay, like this is a bacterial cell, and it has some transporters. You can see this is the transporters. When this metal cells intake in the cell, because of oxidation reduction process, the metal ions get reduced, and it produces different shape and sizes of the nanoparticles. Later I can tell you about how this shape and size are formed, and especially the proteins, the reducers like protein enzymes, cofactors, and reductase enzymes are involved in the synthesis of nanoparticle in case of the intracellular. But in case of extracellular, these proteins are secreted outside the cells, and this secreted or maybe bounded on the cell surfaces. So this can reduce the metal ions to the nanoparticles. So this is just a small mechanism. Okay, so in case we saw the plant, then bacteria, and now time for the algae. So algae is like a omnivorous, like everywhere present. It may be present in the lake, river, or ocean. Mostly in Korea, like people's focusing on the algae. So I also have much more interest about that. So I'm just explaining about short description on using algae as a reducing agent. So this algae can be used directly with the metal salt, and we can produce the nanoparticle, or we can obtain some biomolecules from this algae, and then we can use for synthesis of nanoparticles, or by filtration, the supernatant only we can use for synthesis of nanoparticles, or the biomass can be crushed and we can use for the synthesis of nanoparticles. There are different ways for synthesis of nanoparticle using the algae. Okay. like how these different shapes and size of nanoparticles are formed. So basically, like metal salt mixed with the microorganisms, we can say the extracellular component or supernatant of microorganisms mixed together, or whole microorganisms can be used for synthesis. Then this mixing of metal salt and microorganisms together, and after a particular time of interval, we can say a reaction time. After that, we can achieve a synthesis of nanoparticle. But this is having heterogeneous population of nanoparticles, means different shapes and sizes of the nanoparticle. So the question is like, is it possible to control the shape or size of these nanoparticles? Yes, the answer is yes. We can optimize the process parameters and we can obtain one form of nanoparticle with a specific shape and size. The shape and size is, I means, that is completely depend on the reduction potential of the reducing agent, like proteins or enzymes present here from the microorganisms. They have that ability to reduce. So we can vary the reaction time, temperature, the reaction mixture, the metal salt concentration and microorganisms concentration. If we change, that also make the effect, and the reaction pH. And after that, we can obtain homogeneous capped nanoparticles, which is stable. And it may be a different shapes like a rod, spherical, hexagonal, triangular, or square shapes. Okay, so this is like the synthesis process with
sir, could you start the presentation mode?
Yes, sir. Just 1 minute only.
Okay.
Just 1 minute, sir. Yeah, thank you. Like this, nanoparticles having different shapes, sizes, and we saw the different methods and their applications. We can say this nanoparticles are applicable in the case of medicine, chemistry, and many things in the energy and material science. Okay, so this is all about the basic thing about the nanotechnology. Actually, it's not a part of presentation, but for making interest on the students or the attending undergraduates and graduate students, I just add this portion because I really love to understand the basic things and then can go for the real research. Now here I am presenting my research, which is like the title is like green synthesis of silver nanoparticle by using the Sasa-Borealis. So in this case, in my research, I used the Sasa-Borealis leaf. So ultimately question is like, why only Sasa-Borealis? Because this plant is commonly known as the bamboo, and the plant having many medicinal properties and present everywhere. It is worldwide spreaded plant. So medicinal properties include antimicrobial, antioxidant, and anti-cancer activities. So when the nanoparticles synthesize, they cover the nanoparticles. So ultimately, the nano form of these biocomponents cover the nanomaterials, and they can show a good activity. So for synthesis of nanoparticle using Sasa-Borealis, we use this leaf part of the Sasa-Borealis plant and then boil at 70 degrees Celsius for 1 hour. Then after that, we extracted and we centrifuge at 350 RPM to remove the dead part or unnecessary materials. And after that, we filtered it and then mix with the silver nitrate solution. And by varying different reaction parameters like reaction pH, temperature, and different concentration of plant extract and salt, and we obtain different shape and sizes of nanoparticles. And then next step is characterization and then application. Okay, so we can see the details like how it worked. So for instance, like to know about the synthesis, is this working or not? So we first prepared the plant extract. This is plant extract, then metal salt, and by carrying reaction at different pH. The metal salt solution pH we adjusted with the pH value of 357 9, and 11. And then 1% of plant extract were added, and at 80 degrees Celsius for 30 minutes, we continuously observe the reaction. And then we found the different band peaks. So the absorption peak between 400 and 450 is the characteristics peak of the silver nanoparticles. And then we found like as the pH increases, the synthesis rate is also increases. Because of pH changes, the nucleation is enhanced. And at the higher pH, at the 12 pH, we avoided following this pH because of at high pH, there is high pH and low pH. There is chances of neutralization of biomolecules which are present in the plant extract. So here we observe as the pH increases, the absorption value is increases. This absorption value indicates the amount of nanoparticles in the solution. And as we can see, the reaction tubes color is also changed. The formation of brown color is the indication of silver nanoparticle. Okay, then after that, our second target is like, is temperature affect the synthesis of nanoparticle? So we studied increasing temperature from 40 degrees Celsius to the 80 degrees Celsius, and we performed the reaction at these parameters, like using 1 millimolar silver nitrate solution with 1% plant extract and 30 minutes. So only the temperature were changed. And it is observed that as the temperature increases, the reaction rate is enhanced and the formation of nanoparticle is also increased. So it is concluded that the temperature, it's universal rule, the temperature always accelerate the reaction. So we observe the acceleration of reaction. After that, we check. After that, we check the effect of silver nitrate solution, whether it's a kind of competitive assay. The competitive assay between the silver nitrate silver ions and the reducing agent. So as the concentration increases, the absorbance is increases because of a maximum number of nanoparticles formation. And okay, I have missed one image here. Actually, it should be a plant extract concentration. Okay, so as the concentration of silver nitrate increases, the abundance of nanoparticles is increases. And then at the end, we check the time. The reaction time varies from 5 minutes to the 40 minutes. And we observe that as the time increases, the reaction formation of nanoparticle is also increases. This is our preliminary studies, and from this all different parameters we finalize that the optimized parameter what should be used for the getting homogeneous nanoparticle with maximum production. So it is pH 9, 4% of plant extract, 3 millimolar silver nitrate solution at 80 degrees Celsius, and reaction time should be 40 minutes. And by using this parameter, optimized parameter, we synthesize the nanoparticles, and that nanoparticles proceed for further characterization and the biomedical applications. The synthesized nanomaterials were characterized for the elemental composition, their crystalline nature, their zeta potential, and the morphology. As I told you, by using energy dispersive X-ray with the help of scanning electron microscope, we can know about the elemental composition of nanomaterials. So our synthesized nanomaterial shows energy peak at 2.7 and 3, so which corresponds to the chlorine and silver. So our synthesized nanoparticles having CLNAG, that is confirmed. So whether it is separate or combined together. So for confirming that, we check the XRD, X-ray diffractometer, and from the diffraction peak, it is observed that there are two kinds of crystal elements there. One is silver and second one is silver chloride. So by comparing these peaks with the standard JCPDS files, we confirm that the synthesized nanomaterial is silver and silver chloride nanomaterials. Whether these nanomaterials are small or bigger and which size. So for getting answer, we check the synthesized nanomaterial by transmission electron microscopy, and it is observed that the nanomaterials were mostly spherical oval shape, and they are in the range of 10 to 40 nanometers. So we confirm these nanomaterials. Okay, now we know the nanomaterial is silver chloride, and it has crystalline nature and it has elemental composition, but whether it is stable or not stable. So for confirming that, we check the zeta potential value by light electrophoretic spectroscopy, and we observe the minus 12.95 value. This value indicates that the nanomaterial having negative charges on their surfaces, and because of this negative charges, the nanoparticles were repelled from each other, so that prevents the formation of aggregates or clusters or bigger particles. So we can say this negative charge provide the stability to the nanoparticles. Okay, after that, these nanoparticles were synthesized, but because of what? So for that, we check on the Fourier transform infrared spectroscopy. We prepared the palette of KBR and plant material, KBR and synthesized nanoparticles, and we check the transmission, and we observe that some significant peak variations on different locations. So this peak indicates that the involvement of different functional groups in the reduction process and formation of nanoparticles. For example, this peak is assigned for the alcohols and phenols. The other peaks like amines, alkanes, and aliphatic amines. Mostly the OH group, the alcohol groups are strongly involved in the reduction process, and almost every plant has this kind of phenols and alcohols. Especially the secondary metabolites, they are having strong reducing activity, and whatever the proteins, enzymes, and amino acids, those containing the nitrogen compositions, that is the reducing agents. So from this study, we get know about the biomolecules from this plant material involved in the reduction process, and they were successfully studied by the FTIR spectroscopy. So synthesized nanomaterial were checked for the antibacterial activity. So in our studies, we use 4 different bacteria: the Bacillus cereus and Staphylococcus saprophyticus. This is gram-positive bacteria. And another bacteria is Escherichia coli and Salmonella typhimurium, the gram-negative bacteria. The gram-positive and gram-negative bacteria, they are vary with their cell wall compositions. So mostly we can say gram-positive, gram-negative, almost all gram-negative bacteria are the pathogens. The Salmonella typhimurium is the causative agent of typhoid, as we know. So this is Staphylococcus saprophyticus, especially we can easily found this one on the bread or some kind of wheat flours. We can easily obtain these pathogens. These are the opportunistic pathogens. When the human power, immune power is lower, this pathogen act as a strong disease-causing agent. So by using the agar-well method, we check this nanoparticles activity, and we found that different concentration having different activity on different organisms. as we can see here, 100 microgram per ml of these silver nanoparticles having zone of inhibition. Zone of inhibition is this one, the clear area. That means that area, the organisms in this area are killed because of silver. So it varies with organism to organism. So this is just a qualitative method. So by qualitative, we can say the synthesized nanoparticle having the antimicrobial activity. And then we go for the deeper study, like exactly how much concentration it needs for the complete inhibition and how much concentration it needs for complete killing of microorganisms. So for that, we check broad dilution and colorimetric assay by MIC and MBC. MIC is the minimum inhibitory concentration that kills the visible growth of bacteria that we can see in the microscope. That means in case of MIC, like, okay, it kills the bacteria, but the sporulated form of bacteria that may grow again. So we cannot say MIC is the final concentration where we can completely eliminate the microbial growth. So for that purpose, we must need to check the MBC, minimum bactericidal concentration, where we can say 99.99% bacteria were killed. So for that, we check on the gram-positive and gram-negative bacteria, and we found that different concentrations of nanoparticles having ability to inhibit the growth and kill the microorganisms. So in case of gram-positive bacteria, having a less concentration required for the MIC and MBC compared to the gram-negative bacteria. Because gram-negative bacteria having outer membrane cover with the capsule, the polysaccharide materials, that is prevent the attachment of the materials to the cell wall, and it prevent the inhibitory activity or adverse effect of outside materials. So as we know, like different antibiotics or different multi-drug therapy, as we know, multiple drug therapy, by using different pharmaceutical agent, we can control one disease. This is because the bacteria is modifying themselves and making resistance to the one drug. So at that time, second drug can inhibit that bacteria. And each drug has its specific mechanism for killing. So to observe the ability of silver nanoparticles to kill the bacteria, how it works, so we check the transmission, we check with the transmission electron microscopy, and we found that this nanoparticles damage the cell wall of bacteria and kills. So here you can see the Bacillus E. coli and Bacillus cereus. This is like a control. This is MIC concentration, where MIC concentration can inhibit the cells but cannot kill. So this is double of MIC concentration, where we can see complete killing. And in this case, you can see the cell membrane or the entire structure of bacteria is disturbed. So by formation of blebs or disruption of cell wall, the cell content are released, and because of that, the organism is not able to reproduce energy or replication of their genetic material and synthesis of proteins, which is required or essential for the growth of organisms. And due to lacking of these all things, the bacteria were die. So this is about the antibacterial activity of nanoparticles. And then the second study we perform with the synthesized nanoparticle is the anti-cancer activity. So first thing we have to check is whether these synthesized nanoparticles are harmful to the human being or not. Then only we can proceed for the applications. So for that purpose, we use the skin keratinocyte cells, the name is Hackett, and we check different concentrations, and we found that more than the 90% of viability of cell at the 200 microgram of gram per ml of nanoparticles having cell viability. So up to that concentration, after that, the cell viability will decrease. So we didn't use that concentration. So we check up to 200 microgram only. And then these nanoparticles were tested on the gastric adenocarcinoma cells, the AGS cells, and we observe the concentration dependence or dose dependent decrease cell viability were observed. So this decrease were concentration dependent and 24 hours. It may vary with the time also. This is 24-hour study. So we observe different concentrations and we calculate the IC50 of these nanoparticles. IC50 is the concentration where we can say 50% of cells are killed. Even doctors, whatever injections or whatever doses preferring us is having the 50% potential to treat you. That's why we need the multiple injections or multiple drugs for curing one kind of disease. So this is the like basic funda behind using IC50 concentration. So, and for checking viability, we usually use the 96 well plates, and by using the colorimetric agents like MTT, that agent, this is MTT agent. This MTT agents having ability to interact with the adenine and thymine of the live cells, and it shows the conjugation and shows the color formation. And by using the colorimetric spectrophotometer, we can check how much percentage of cells are alive or dead. And there are some other techniques also, but this is just a quantitative method, so we prefer the colorimetric assay. And for knowing about the mechanism behind the anti-cancer activity of these nanomaterials, we took a step forward, and that is the immunofluorescence staining. So the genetic material, that is DNA of these cancer cells, were stained with the different staining solutions, and we check the effect of these nanomaterials on the cancer cells. So by using crystal violet, you can see the lot of blue spot. This is like a live cell nucleus. So these are like control where no nanoparticles were treated. And at the same time, the IC50 concentration were treated, and you can see the numbers are reduced. So these numbers are reduced, but how exactly the cells were died or killed by these nanoparticles? So for that purpose, we check with the DAPI solution. This is an immunofluorescence stain that binds with the adenine and thymine 80 content of the DNA, and it gives the blue fluorescence in the confocal microscopy. So in case of control region, you can see this is the entire spherical oval shape of nucleus because of no damage in the cells. And like some are, you can see uneven nucleus because the cells are growing, maturing, and they are dying by a systematic death pathway that we call as apoptosis. So this is like entire things. But in case of the nanoparticle treated cells, you can see this formation of fragments or blebs or like disruption of the nucleus. So we can say the nanoparticles were interacted with the genetic material, and they disturb or they cut the synthesis pathway, and they responsible for the death of these cancer cells. So in short, we can see the different, this is just one pathway we saw in our study. So further studies are ongoing. So on at protein level and genetic level. So very soon we can have that breakthrough, like how these particles acting as an anti-cancer agent. So schematically, we can see here like how these nanoparticles having antibacterial and anti-cancer activity and their mechanisms. So these nanoparticles, they are acting as an antimicrobial agent. At that time, the bacteria don't have the cell wall, the cell membrane, the bacteria have cell membrane, which is protected with the different proteins. So these nanoparticles are transported inside the cell membrane, and they are responsible for formation of reactive oxygen species, and this can damage the proteins, genetic material, DNA, or some enzymes, and they, because of dysfunctioning of these proteins or enzymes, the replication process is stopped and translation cannot occur, and at the end cell cannot divide by natural way. And another reason is like this nanoparticle increase the cell permeability, so the cell content can be drained off from the cell, and because of lacking this cell function, the cell cannot survive. This is about the antibacterial thing. And in case of the anti-cancer, this nanoparticle can go to increase the reactive oxygen species, and which cause harm to the proteins and enzymes, and by going through different pathways, it can harm to the mitochondria, and then caspase activations. The caspase pathway is an important pathway to regulate the proliferation of the cell in the cell biology. So they deactivate or activate the caspase activity like pro and pre. So activation is harmful and deactivation is also harmful. So at certain level, it causes deactivation. And because of all these reasons like enzyme disruption, DNA damage, protein denaturation, and the activation of caspase, it goes the systematic death, that is called apoptosis. And at the end, we can say the cells are dead. So this is just a schematic presentation, and you can understand from this like how this nanoparticle acting. So this is all about our research, what we had done. So in summary, I can say we synthesized the silver chloride nanoparticle by using the Sasa borealis leaf extract, just by using water extract. It's eco-friendly, cost-effective, one-step, and facile. And the synthesized nanoparticle were very small, like a spherical 10 to 30 nanometer, and their zeta potential indicates the negative value, which provide the stability of to the nanoparticle by electrostatically repulsion. And then on the basis of different techniques like EDX, XRD, we check the crystalline nature of the nanoparticles, and the synthesized nanoparticle where we check for the antibacterial activity, and we found that they are more effective on the gram-positive bacteria, as well as they are effective on the gram-negative bacteria, but they have more potential to kill the gram-positive bacteria. And this synthesized nanoparticles were at certain concentration non-toxic to the human cells, human skill cells, while at the same concentration, below the same concentration, it shows the concentration dependent cytotoxic activity. And these nanoparticles were found inhibiting the DNA structure, damaging the DNA structure, and leading the death of cancer cells. So this is all about my presentation. I hope this is useful for many of you. And then I will ask you, like, do you have any question, please?
Very nice, Dr. Mahesh. Now the session is open for the discussion. If you are having any questions, you can ask participants. Dr. Mahesh, I have one question.
Yes.
Especially, am I audible? Can you hear me properly?
Yes, yeah, sure.
You have explained the one of the case study, that is synthesis of silver nanoparticles using Sasa borealis bamboo plant. And you mentioned that it has got a wide applications in antibacterial and especially anti-cancer activities. So in the summary, you mentioned you have synthesized silver and silver chloride nanoparticles.
Yes, sir.
Two phases it has shown. Why not only the silver, or why not only the silver chloride? Why mixed phase is observed?
Actually, because of presence of chloride containing component in the phyto extracts, so it was synthesized as a silver chloride. but as we have other plants and we successfully synthesize silver separately. But in this case, we had silver and silver chloride.
In this
So as
case,
we know, like chloride is.
can we have any other remedy that we can separate these two?
For separation, I don't know exactly, Professor, how it will work. But as we got with this plant extract, and the same we were reported in our studies.
Okay.
So we didn't targeted for separation. It was my PhD work, so I just reported and I finished my studies.
Okay, and related with the same, you have explained very well how can we generate the different shapes and size of nanoparticles by controlling the reaction parameters. In this case, particularly in this case, by the bamboo plant, that silver, because in summary you mentioned that you have got the spherical nanoparticles.
Yes, sir.
But is it possible to get some other shapes in the same case?
Yes, sir. Yes, sir. Actually, in case of Maduka indica, the Mahu plant in our India, we call Mahu. We use that flower extract. So when we just use that flower extract and synthesize at room temperature, so we got heterogeneous, like triangular, spherical, oval, like that shape. So and when we go at higher temperature, like hydrothermal method at 180 degrees Celsius, we successfully synthesized the hexagonal nanoparticles, actually nanoplates that were about 220 nanometers size.
Very good.
And that is really having good effect on the sunlight radiation reflection. That is nowadays the trend is going on the smart window to maintain room inside temperature to repel the outside rays. So we are targeting that nanoparticles for coating the window glasses to maintain inside cool temperature to avoid the outer temperature in the room.
Here, negative zeta potential related with that, you quoted one line, it provides the stability, maybe to prevent the agglomeration or a cluster. But in some cases, I have seen some report where even the negative potential, some agglomeration is also observed. Is there any other reason because of which stability or only the negative zeta potential?
No, sir. No, sir. This is just a kind of one aspect we can say. There are, as we know, like it's a chemical and every chemical has its own properties. So we can stabilize by using different polymers. And just with the view of electrostatic repulsion, I can say it will provide the stability.
Very good. Very good. Very good.
Thank you.
Other questions?
Sir, I would like to ask one query in which I'm unaware about that. So, sir, could you please elaborate on biofilm biofilm inhibition process or that biofilm formation or like that?
Yes, sir. Actually, what is the biofilm? Biofilm is the film produced by the microorganisms over the hard surface. Especially, this is a major issue with the pharmaceutical or nutraceuticals or food industry, where they are using the continuous water flow with the stainless steel pipes, and they are facing like on the storage tank also, they are facing the formation of slime layer, sticky layer. That is because of the pathogens and bacteria. Bacteria have ability to produce extracellular material that is mostly polysaccharides, and because of that polysaccharide, it creates a stick to the metal surface and it creates a layer, and it is having ability to cause endanger anytime. So to remove that layer, we usually use the silver nitrate and hydrogen peroxides and some hypoxic materials. So silver nanoparticles is one kind of, but I cannot recommend to say like it's really useful, because of nanoparticle can be after treatment, like nanoparticle can be get discharged to the environment, and it will have some adverse effect. And disruption of cell membrane, as I told you, the nanoparticle interact with the cell membrane or cell component like DNA, proteins, enzymes, and it increase the cell permeability and lose the cell content. And at the end, we can say empty of the cell, that is killing of bacteria. And in this way, we can remove that biofilm. But it's not that much easy what I say. This is just one point of view.
Thank you.
Sir, I have one question.
Yes, sir.
Sir, can bacteria develop resistance to nanoparticles?
Of course. Actually, bacteria are smarter than any others. Usually, they are, as you remember, like penicillin, like decade ago, penicillin were the one drug which can be used for any kind of diseases. But nowadays, it's not possible to resist the disease by using penicillin, because microorganisms, they are adopted, they change their cell wall, their receptors in such a way that they cannot recognize that. And in case of nanoparticles, it is possible, I can say, because of the nanoparticles are transported inside the cell, or they can be attached on the cell by recognition of receptors. So it may possible bacteria can change their receptors, and the gram-negative bacteria, they have ability to produce the extracellular layer, that is capsule. So by producing that slimy layer or capsule, they can easily make resistance to these nanoparticles.
Okay, thank you, sir.
Is there any question, or shall I conclude, organizers?
Yes, sir. You can proceed, sir.
Okay. Thank you, Dr. Mahesh. Your topic, you started talking on the metallic nanoparticles, the green synthesis, and their biomedical applications. I think this is a very important topic, not only for chemistry, for all part of science, because in particularly under SPPU, the syllabus of nanoscience and related is started from SYBSc, I think, for chemistry as well as for physics, electronics, and even zoology students. And for all the corners of that science, this topic is very helpful for UG as well as for the research students. So you explained all the general aspects of green synthesis, the mechanism of that synthesis using plant extract, using microorganisms. You have explained all the three methods, that is a chemical synthesis, physical synthesis, and biological synthesis for nanoparticle development. The very important under microorganisms, you explain the intracellular biosynthesis and extracellular biosynthesis and the role of enzymes in the reduction of nanoparticles, particularly in both the cases of you explain how the formation of different shapes and size and what is the importance of different shapes and size by controlling the reaction parameters, because particularly in the case of nanoparticles, this shape and size is very important. And by giving one example of synthesis of silver nanoparticles as a case study by using the bamboo plant, even by using algae, you have explained all that aspects, and how the developed nanoparticles, silver nanoparticles, are having applications in antibacterial and particularly in anti-cancer activities. This is very important. Definitely your lecture is helpful to the researcher as well as UG students for the developing their approach towards the nanotechnology and the study of nanoscience. I am very much thankful on behalf of organizers, Dr. Mahesh, that you have given a very nice lecture. Thank you. Thank you.
Thank you, sir. Thank you. Thank you. Thank you so much.
Thank you. Thank you, Patil, sir. Indeed, it was a splendid lecture, and I think every participant must have enjoyed the lecture. Now, I would like to request Professor Sadgir Madam to propose the vote of thanks for this session. Over to Sadgir Madam.
Good afternoon, one and all. I am Dr. Sadgir. I feel honor and privilege that the technical session. On the behalf of our college principal organizing committee, I express heartily thanks to today's resource person, Dr. Mahesh Kumar Patil sir. In his talk, he covered about the green synthesis of metallic nanoparticles, their antimicrobial and anti-cancer activity. And I would also like to thank today's chairperson, Principal Dr. A.V. Patil sir, for the nanoparticle synthesis, their characterization, their biological activity. I would also like to thank the participant for participating in today's session. Over to adoration.
Thank you, madam. Thank you, sir, for sparing your valuable time and guiding all the participants and dear students who are participating in this international conference. Thank you, sir.
Thank you.
Now, with a break of 10 to 15 minutes, we are going to start a second session. And in the second session, Professor Dr. Avinash Kumbar sir from Savitribai Phule University is going to deliver his talk. So, dear participant, we are going to meet again here only at exactly 1:50 p.m. So, you are requested to remain present here via this online mode. So don't go anywhere. We are going to start this second session within 15 minutes. Okay, thank you. Sadgit madam, it's over to you now. You are going to take the charge for the second session.
Okay, sir. Thank you, sir. Next session will start at 1:50. All are requested to join the second session.
Resource person
Sir, could you stop the screen sharing? Mahesh Patil sir. Thank you, sir. Madam, Kumbar sir, I think Kumbar sir has joined. Sadgir madam.
हा सर एकच मिनिट.
सर मी जॉईन झालोय.
Thank you, sir. Thank you very much. Welcome, sir.
सर नमस्कार.
नमस्कार नमस्कार.
योग येतोय असं भेटण्याचं ऑनलाईन आम्हाला बरं वाटतं. युनिव्हर्सिटी आणि मान्यवर त्याचे तज्ञ आम्हाला ऑनलाईन भेटतायत आणि कुंभार सरांचं तर दर महिन्याला आपण भेटतोय. त्यामुळे बरं वाटतं.
येस सर. म्हणजे सर नमस्कार.
नमस्कार नमस्कार. एक मिनिट.
सर
Yes, madam, you can start.
झाले सगळे जॉईन.
You may start now. बनगे सर नमस्कार. भालू बोलतोय कोणाशी बोलतोय काय माहिती.
हॅलो.
हा
नमस्कार.
नमस्कार.
नमस्कार नमस्कार.
अविनाशजी पण आहेत. आमचे सन्मान्य प्राचार्य आहेत. मॅडम सदगीर मॅडम.
हो सर सुरू करते. यस सर. चला स्टार्ट सर.
Yes, you proceed.
Respected dignitaries, the total development of the country is depend on the quality of higher education. Education, training, research, and community service are all part of higher education's goal and vision. It is an effective instrument for constructing a knowledge-based society. Quality research is required for any educational institution to progress. Keeping all of these in mind and with the help of the guidance of Honorable Principal Dr. P.S. Jagdale Sir, IQC and BOS Chemistry MGB, the Department of Chemistry of our college has organized these international e-conference on current research in chemistry and nanoscience 2022. Welcome back again for the second session. I welcome you all for the second session of the international conference. Ladies and gentlemen, we are having a great opportunity to listen to the words by great knowledgeable speaker, Professor Dr. A.B. Kumbar Sir from Savitribai Phule, Pune University. I would also like to welcome Professor Dr. B.M. Bange Sir, Institute of Chemical Technology, Mumbai, for chairing the session. Now, I would like to request Mr. S.A. Aire Sir to introduce Professor Avinash Kumbar Sir and Professor Dr. B.M. Bange Sir. Over to you, sir.
Thank you, ma'am. Good afternoon, everyone. This is Satish Aire from Department of Chemistry. In this very special occasion, I am here to introduce the speaker of this session, Professor Dr. Avinash Kumbar Sir. He is working as a professor in Department of Chemistry, Savitribai Phule, Pune University. Sir has completed his BSc in Chemistry from University of Pune in 1985 and MSc in Inorganic Chemistry from University of Pune in 1987. Sir has received PhD in Bioinorganic Chemistry from the same university in 1993. Sir has received postdoctoral fellowship from University of North Carolina, Chapel Hill, USA in the year 1998. Boyscast Fellowship from Department of Science and Technology, Government of India, Fulbright Nehru Senior Research Fellow, University of Houston, USA. Sir has received CRSI Bronze Medal in 2014 for his contributions to research in chemistry. Sir has received Journal Grant for International Authors by Royal Society of Chemistry in 2013 from National University of Ireland. Sir is a fellow of Maharashtra Academy of Science. Sir has received
Professor Arun K. Day Award for the paper presentation at the Convention of Chemists in 1995 by the Indian Chemical Society. In his profession, Sir worked as a coordinator for novel materials thirst area under UPE program, SPPU Pune, coordinator for University Science Club, coordinator for Center for Advanced Studies in Chemistry. Sir also worked as a coordinator for BSc blended course, Savitribai Phule Pune University in collaboration with University of Melbourne, Australia and ICER Pune. Sir worked as a coordinator for NCFRR 2005. Sir is a work as a course coordinator for GIAN course on advanced organometallic chemistry in 2016. Sir is a council and life member of Indian Society for Radiation and Photochemical Sciences. Sir worked as a subject expert on different committees. Especially, sir has a research interest in metal complex DNA interactions, metal organic frameworks, pulse radiolysis. Sir has published 69 research articles in highly reputed journals. Among these, 7 of the journals are elsewhere and Royal Society of Chemistry. For this session, we also have the chairperson, Professor Balchandra Bange sir. Sir is working as a Dean, Infrastructure and Campus Development and Professor of Industrial and Engineering Chemistry, Department of Chemistry, Institute of Chemical Technology, Matunga. Sir completed his BSc in 1986 from Abasaheb Garware College, Pune and MSc from Department of Chemistry, Savitribai Phule Pune University. Sir has qualified CSIR NET exam twice in 1988. Sir has received PhD in 1996 from Savitribai Phule Pune University. Sir has received JSPS Fellow in Tohoku University, Sedan, Japan, Crest Fellow in Hokkaido University, Japan. Sir worked as a coordinator for Marathi Encyclopedia for chemistry subject, Maharashtra State Marathi Encyclopedia, Production Board, Government of Maharashtra. Sir has published 391 research articles in international journals. Sir worked as an editor of 2 books and author of 20 book chapters. Sir has filed 13 patents and 21 patents on his name. Sir has received many awards such as Best Paper of the Year Award in 1995 by NCL, Patent Award by NCL Research Foundation, Bronze Medal for Contribution in Research by Chemical Research Society of India. In his profession, Sir worked on different committees such as Advisory Board Member of Royal Society of Chemistry, Associate Editor, Catalysis in Green Chemistry and Engineering, Secretary of ACS India International Chapter, Chairperson of Catalysis Society of India, Mumbai Chapter. Sir is a life member of Biotech Research Society India. He is a recognized PhD guide for the chemistry, biotechnology sciences, and chemical engineering. Sir has completed many research projects funded by Department of Science and Technology, Department of Science and Technology under DST JSPS Indo-Japan Project, Department of Biotechnology, All India Council for Technical Education. Sir has published 391 research articles in highly reputed journals. Now I request Professor Balchandra Bange sir, please chair this session. Over to you, sir. Thank you all.
Dear esteemed panelists, it's my privilege to chair this session. And we are here for today lecture by Professor Avinash Kumbar, and just now we have introduction of Professor Kumbar, who has done great contributions in the field of inorganic chemistry, and he has significant contributions. So I request Professor Kumbar to start his talk in this session.
Thank you, sir. Thank you, Bhange sir, for this introduction and for chairing this session. It's a great pleasure to be on this platform. I would like to thank all the members, Professor Principal Jagdale sir, Kapadnis sir, and all faculty of LVH College for giving me this opportunity to speak to you and on a topic which is very dear to all of us. And surprisingly, Bhange sir is the chairman today. The topic is related to catalysis, and so I will like to share my screen and then start. Is it? Is the screen visible?
Yes, sir. Yes, it's quite visible.
Yeah, and I am also audible, right? So the topic I have chosen today is water splitting by transition metal polypyridyl complexes, because that is the need of the our energy is a major thrust area for all of us, especially majority of the chemists and material scientists are working in this field for trying to generate energy, and hydrogen and oxygen from water is one of the cheapest source of energy that we can think of. So to begin with, see if I can shift the next slide. So why cannot I share? I'm not able to. Yeah, the sequence would be that I would give a brief introduction followed by experimental techniques that are used for this kind of research. And I'll talk to you about mononuclear ruthenium polypyridyl complexes, which are the ones that are used for homogeneous water oxidation. And I would also follow it by trying to synthesize a diode assembly for light-driven hydrogen evolution, and then finally summarize the topic. So obviously the motivation for all this comes from several initiatives that everyone is trying to take for to understand how we can utilize energy, or how we can utilize cheap energy. And there are several aspects, like for example CO2 mitigation is one of the topics, and we have a lot of melting of ice, and trying to see whether we can have alternate energy sources for a generation of energy and utilization of energy, which will be environmental friendly. In that aspect, obviously we are trying to copy from nature, and natural photosynthesis is the process that we are all trying to mimic, which basically uses two schemes, as we call them, as PS1 and PS2, wherein the plants as well as some bacteria can convert energy, or can convert H2O to O2, followed by, which is preceded by a sequence of electron transfer events coordinated or catalyzed by several groups, which include several cytochromes, plastoquinones, and finally leads to conversion of H2O to oxygen and hydrogen by the complex called as the water oxidation complex. And that the overall process is conversion of carbon dioxide and water to carbohydrates and oxygen. Plants do this, as you know, in a very, very efficient manner. However, we are trying to copy that. The reason for copying that is these plants have a very stable structure. The mechanism or the structure that is present in which plants do that is very stable. It's a very high turnover frequency of about 300 to 400 second inverse. The turnover number is about 1,80,000 and it can regenerate within half an hour. This is catalyzed by a manganese. The water oxidation is catalyzed by a manganese complex in nature, which has a very intricate structure. It is a tetranuclear manganese structure and has calcium as one of the centers which supports this water oxidation. And so therefore many people are trying to mimic this, and majority of them are trying to use ruthenium complexes to generate both oxygen from water and also to convert the 4H plus generated to hydrogen, which can be used as fuels. Process this process. Whereas in normal natural photosynthesis, we know that this process is carried out by a manganese, as I mentioned before, and this can be done by light driven, which is using sunlight. So therefore, we are trying to develop nature and also generate oxygen and hydrogen. Yeah, the process is one second. I'm trying to. Yeah, the water oxidation or water splitting, as it is called as, is as you see here, conversion of H2O to oxygen and 4H plus and 4 electrons. Is about the potential that is required is about 1.23 volts, and it also involves the proton or water reduction, conversion of 4H plus to 2H2 with an E0 of 0. There are several ways in which this water splitting has been carried out. Some of the simplest ways are using chemicals, sometimes light driven, electrocatalytic or electrolysis, and also thermal, radiolytic, as well as photobiology. These are some of the ways in which water splitting can be carried out. If we go only to the water oxidation, that is conversion of H2O to O2, requires ideal catalyst. Obviously, the ideal catalyst should overcome the barrier of 1.23 volts. It should easily prepare. It should be low cost, stable at a long range of pH, and it should have a very high turnover number, which should be efficient catalyst. The water oxidation catalysts that are reported up till now in literature are obviously metal oxides, in some cases metal organic frameworks, polyoxometallics, nanoparticles, metal complexes, and also some hybrid materials. I would not go into the details of the reactions at the moment. We can do that later. But the methods that are employed for this are chemical water oxidation, light-driven water oxidation, and to find out or to estimate, to find out the turnover number or the turnover frequency, and also to calculate the rate constant and compare it with the other systems to try to find out the most efficient systems that are used for water oxidation. Coming to the homogeneous water oxidation, because we are going to restrict to that, these are mainly catalyzed by transition metal complexes using ruthenium polypyridyl complexes, and several ruthenium polypyridyl complexes have been tried and used as water oxidation catalysts and have been classified into 4 types, such as ruthenium polypyridyl complexes, which are termed as type 1, type 2, and type 3 catalysts. Here are some of the type 1 catalysts that are being used. These are simple polypyridyl complexes with ruthenium as the metal and have a variety of ligands that are attached to that. In the type 1 catalyst, you can you have ruthenium polypyridyl complexes with other ligands. In the type 2 catalyst, we also have several ruthenium polypyridyl complexes with water coordinated. And as you can see here, we have type 3 catalysts which have polypyridyl ligands with coordinated water molecules, or in some cases you have pyridines which are coordinated, which can be replaced by water. So either we can have a precursor or we can have
कुंभार सर,
हाँ सर,
तुमच्या पाठीमागनं जरा जास्त आवाज येतो म्हणून तुमचा आवाज स्पष्ट.
आवाज येतोय?
हो, पाठीमागनं कोणतरी म्हणजे डिस्टर्बन्सेस. तुमचा माईक बंद झालाय सर, कुंभार सर. Kumbar sir, you are muted yourself. Hello, Kumbar sir. Hello.
हाँ सर, आता येतोय?
हाँ सर, तुमचा आवाज येतोय क्लियर.
Yeah, so we have several such complexes which can be used as
your video is off, sir.
Catalyst.
Your video is off.
Okay. Yeah, which are used as catalysts,
yes,
which can be used as catalysts. And Thammal et al. Have actually classified, and several such compounds they have prepared. There is a lot of electronic and steric nature of the complexes that is used or that is made use of, and which helps in improving the catalytic activity of these complexes. So to give you a brief idea, that synthesis of these complexes can be quite simple, and we can add additional factors such as co-ligands, which will help in catalyzing or modifying the electron center by changing the electronic properties of the donor ligands, and it can also help by ancillary ligands which might sterically help the coordination of water or any other ligand for carrying out this catalytic reactions. So what we are trying to do is, by derivatizing these ligands can help in affecting the ruthenium center, metal center in this case, by electronical effects as well as by steric effects, by modifying the R groups in this case, such as here we have a sulfato group that is attached over here, which helps, or hydrogen bonds with this water molecule, to keep them coordinated to the ruthenium and therefore help the catalysis. So several modifications can be done on these ligands, and there are several groups that work on the modification of these ligands, and therefore they help in the catalytic activity. In general, the most accepted mechanism for such water oxidation is what we call as one of the mechanism that is called as water as a nucleophilic group. So when we start with this complex, like for example, you have a ruthenium polypyridyl complex, which has ligands such as terpyridine. Okay, I can use the pointer. Yeah, you can use the ruthenium bipyridine, ruthenium terpyridine complex with a water coordinated. And when we are doing oxidation by using a chemical oxidant such as cerium in this case, so we can use cerium as an oxidant. Cerium is one of the best known oxidants for this because we have to a potential that we need to reach is about 1.24. We can start with a cerium oxidant, which gets reduced to cerium 3+, but in turn oxidizes the ruthenium to several ruthenium 3, 4, and in some cases ruthenium 5. So as you can see here, this complex can, with coordinated water molecule, can be oxidized, can be oxidized by one electron oxidation processes. Yeah. Sequential one electron oxidation can convert this ruthenium 2 into ruthenium 4, ruthenium 5 complex, and further addition of H2O will give rise to such peroxo linkages, and this peroxo link will convert to oxygen, and with the addition of water molecule give back our catalyst. This is the most accepted mechanism, as several intermediates have been identified, isolated, and also characterized by spectroscopic as well as some structural techniques. There is another mechanism that is proposed, which is called as an I2 mechanism, wherein it is proposed that the ruthenium water coordinated complex with the terpyridine bipyridine, which has been eliminated here, which has not been shown over here, but again can give on sequential oxidation gives rise to ruthenium 3, ruthenium 4, like in the previous case, but two such molecules come together to form a peroxo linkage in such a way, and with this then combines together to form oxygen. This is a second kind of a mechanism that also has been proposed, but most of the people believe that the first mechanism, or water as a nucleic acid mechanism, works in majority of the mononuclear complexes. So these mechanisms are quite well studied. So knowing that, we have also been working on the ruthenium polypyridyl complexes, and based on that, obviously, we also do a lot of modifications for the complexes. So we began with synthesizing ruthenium polypyridyl complexes using the same terpyridine bipyridine structure, but modifying the bipyridine structure by groups such as what we call as glycouryl groups can be easily synthesized to form this complex. So by modifying several parts of this, the inner coordination sphere, the outer coordination sphere, wherein hydrogen bonding can take place, by changing this structure or by changing this ligand by using phosphine ligands to stabilize the ruthenium structure, whether we can convert water into oxygen, and to see how it compares with the well-known parent complex called as the ruthenium terpyridine bipyridine complex. So we had several samples have prepared, and we have prepared complexes. We will not go into the water reduction, and several, as inorganic chemists, as Vange sir mentioned, we prepare complexes and study them by several techniques such as NMR, H1 NMR, C13, infrared spectroscopy, UV visible spectrofluorescence, time correlated single photon counting, because it involves proton coupled electron transfer, elemental analysis, in some cases mass spectrometry, single crystal X-ray, gas chromatography to generate our oxygen tank, mass spectrum, electrochemistry, because this is a proton coupled electron transfer reaction. And then we have recently reused a pressure transducer or a sensor which actually measures the oxygen or hydrogen gas that is generated, which we can measure continuously, and based on this we can actually identify. So one of the first systems that we studied was the ruthenium bipyridine glycol-uryl complex for photocatalytic and chemical water oxidation. So our forte or our speciality is this ligand, which we call as bipyridine glycol-uryl. And as we all are trying to synthesize this from simple ligand or simple precursors, this just needs phenanthroline and urea as the molecules, and so therefore we can prepare this, and we have several complexes, hundreds of complexes which we have prepared using this ligand. The specialty of this ligand is it has 2 hydrogen bond acceptor in terms of oxygens and 4 hydrogen bond donors, and this can hydrogen bond with other groups in the vicinity and generate a very stable structure, we thought, which would be similar to what calcium and other ligands do in the actual natural water oxidation complex. And calcium, as you can see, there are several, if you see the water oxidation complex crystal structure, in addition to the manganese tetrameric cluster, it has calcium and other groups that hydrogen bond, and that's the reason why we thought that hydrogen bonding and hydrogen acceptance of the bipyridine glycol-uryl helps or can help in stabilizing the structure, and because of the stabilization of the structure, whether it will also help in improving the turnover number and turnover frequency. We have synthesized this complex in the laboratory several times, and very interesting synthesis, gives very good yield. And one of the classic examples of this, one of the best reasons why we actually prepare metal complexes of glycol-uryl is because of the hydrogen bond donor and hydrogen bond acceptor kind of included in the system. It gives a lot of single crystals. It gives crystals, and which we have analyzed by X-ray crystallography. 90% or 95% of the complexes prepared by using bipyridine glycol-uryl have given us crystals and crystal structures. So we are very sure of what the structure is. As you can see here, here is the crystal structure of ruthenium terpyridine BPG, as we call it, bipyridine glycol-uryl, with a chloro ligand. When we dissolve this complex in acetone or acetonitrile with a bit of water in it, we get a crystal structure of that chlorine is being replaced by water. So we have a structure, we know that the crystal structure contains water. We have characterized it by mass spectrum. So we know the exact structure of a chloride and then water. We have characterized them by simple absorption spectroscopy. So it has a typical ruthenium bipyridine or ruthenium polypyridine like structure with a very strong MLCT at 500 nanometers, as you can see here, at 500 nanometers, as you can also see here. As you can see here at 500 nanometers, at 495 nanometers, with a very high, which is typical of piperidine glycoluridine or ruthenium polypyridyl complexes. So we can prepare these complexes. Go next. Yeah. We have characterized these complexes by several techniques that we need to, like for example, we have also characterized them by varying the pH and measuring the absorbance spectrum. And we have found out, based on this, we can find out the pKa of the ligands. Okay. So the pKa of the ligands, as there are protonable groups, including at water, we find that in some of the complexes that values are around 9 and 11.3. In our case, the values are 9.6 and 10.3. So we know that because of the water that is coordinated, it has a pKa value of around 10.3. That means this complex is quite suitable for measuring the water oxidation, or as a suitable candidate for water oxidation. Simple experiments which can be done in the laboratory without much of a infrastructure that is there. And we have also done some absorbance studies by adding have oxidized that ruthenium-3 and try to measure the changes that take place in the absorbance spectrum, because as you see, the ruthenium-2 and ruthenium-3 species have different absorbance peaks. And you can see here that as we sequentially go on adding cerium-4 to the system, it goes on decrease at some certain positions, at certain wavelength, it goes on decreasing and increasing in certain situations. We have also done the same for further when ruthenium-3 gets oxidized to ruthenium-4. So we have identified that in our complex also the sequential oxidation of ruthenium-2 to ruthenium-3, ruthenium-3 to ruthenium-4 is possible. And we have also measured that by changing the pH of the system and plotting a graph of the redox potential versus the pH, which is known as the Pourbaix diagram. And using that Pourbaix diagram, we can actually identify that in this complex, proton coupled electron transfer can take place at various pHs. And so therefore, this complex is a good candidate for measuring the water oxidation. And in fact, to assemble or to draw a Pourbaix diagram is a very difficult proposition. It needs a lot of specific calculation, and which we can do very easily in our lab, and we can actually find out at which regions or which potential this complexes can be utilized as a water oxidation complex. So we have drawn the Pourbaix diagram, and it's quite interesting, and we find that we are becoming nearly experts in drawing the Pourbaix diagram. So we found that, okay, our complex seems to be suitable for use as a catalyst for water oxidation. But we wanted, previously people use this use these complexes, and the oxygen that was generated by after addition of cerium was usually done by gas chromatography. So at every hour we used to with the help of a syringe take out the gas that is generated and measure it by GC, which was quite tedious. But such a simple pressure transducer is available where you can actually measure continuously the gas that is generated. And so therefore we got this simple transducer. We have 2 or 3 of them now. We can use them in a series and also slightly modify it by attaching lights surrounding that, which will also help us. In addition, I mean, we do not have to do a chemical oxidation, but we can also do a photochemical oxidation. So when I attach LED lights on this, wherein it results in a flux, in a light flux, hitting on the complex in the solution, and the oxygen can be measured, or the gas that is generated can be measured. So this is the and it continuously of the gas generated, and we can plot a graph of the amount of gas evolved with respect to the molar concentrations of the complex that we have added. And so therefore that will help us in calculation of the turnover numbers and turnover frequencies for these complexes. So that this is exactly what we have done this system, and as you can see, we have plotted a graph of with respect to time with the oxygen that is evolved. We have analyzed the oxygen by GC and identified it as oxygen. And as you can see here, by changing we have prepared the several complexes having chloride, bromide, iodide, astronitrile, as well as water coordinated, and we find that the turnover numbers for these complexes is 678, 650, and 956 in the case of wind water. And I've also found out the rate constant of these by way of various complexes. So this is an interesting system. As you can see here, we have even other complexes, and if you see the turnover numbers are about 133, 833 or 678 as we go on changing. This is this is what we do. We also do photochemical water oxidation and light driven water oxidation by this complex. So that means we instead of adding cerium, we add light. Within the system, we have a sacrificial oxidant such as sodium thiosulfate, and we measure the water oxidation, and we find that this complex can also do the light-driven water oxidation. So both chemical as well as light-driven water oxidation can be carried out by using these complexes. And when I compare the value of the systems that we have, this is the parent system, which is ruthenium terpyridine bipyridine, which is 178, the turnover number for the systems where we have water and where we have iodine is about nearly 4 times more than the parent system. So this is a good system where we can actually see that most probably the water oxidation is enhanced because of the bipyridine glycol-urine ligand, which helps stabilize the structure more. Then obviously, as you can see, when one system works, other systems will also work and do sort of a structure activity relationship, modification of the ligands that we do, and we have modified these ligands by using other polypyridyl ligands or other polypyridyl ligands with some say sulfur, with sulfoxide based ligands. We can just go on modifying these ligands and try to measure the water oxidation to understand how the modification of sulfur or any other groups over here affects this electronically or sterically, and to see whether there is an effect on the water oxidation to measure turnover numbers. And as you see here, we have the structures of all these complexes is known, so we are very sure about the structure. And we have some more ligands over here, where instead of that we have a diazaflurinone ligand. Again, the structure of that is known. So we are very sure of the structures, and we have done all characterizations that are required to establish the structure, such as absorption spectroscopy. We have done the electrochemistry. We have done the sequential titration with cerium to know whether this complex does oxidize to ruthenium 2, ruthenium 3. We have also estimated that by spectroscopy. And again, the Pourbaix diagram was drawn. And similarly, using the pressure transducer, the water oxidation activity was found out. And as you can see here, by changing the groups from bipyridine glycol-urine to aza or oxo groups, the water oxidation or the turnover number decreases from 800 to nearly 371, 281. So at least a good idea whether these complexes can be used by other substitution gives rise to 602. and turnover numbers were calculated, turnover frequencies can be calculated. So it gives us a good idea of how substitutions to the ligands can give rise to changes in the turnover numbers and frequencies, and we can do a structure activity correlation. So this is the second series of complexes that we had prepared, and compared that with the previous one. Still, with the parent ruthenium terpyridine bipyridine, it still gives rise to good water oxidation. May not be as good as the previous case, but still it has a better water oxidation turnover number. Then the last part of the system that we have is whether it is possible to assemble both the oxidation as well as reduction catalysts to generate in a system or in a combination, what we call as a diode assembly. So several people were using what is known as a very simple cobalt oxime complex as a hydrogen evolution catalyst. What we thought was, can we do this with the help of light, and can we prepare a complex wherein it has both the photosensitizer, that is the ruthenium complex, and a hydrogen evolution catalyst, that is a cobalt oxime complex, within the same molecule. And then by shining light and using sacrificial oxidants, whether it can catalyze the conversion of H2O to hydrogen. So we tried to see whether such an assembly can be synthesized, and we developed that molecule. So these are called as proton reduction reactions or proton reduction catalysts. Again, as we did in the previous case, requirement for an ideal catalyst, again, stability in a long range of pH, easily available, obviously lower cost, and stable in an oxygen environment. There are a few proton reduction catalysts that have been reported. Again, metals, metal oxides, organic frameworks, polyoxometallites, and some nanoparticles, and some hydrogenase enzymes in terms of bioinorganic chemistry have also been used as proton reduction catalysts. But not much was known about metal complexes as catalysts, except for cobalt oxime and a few catalysts, and whether we can combine the catalyst and photosensitizer. Again, to proton reduction catalysts, people have also used homogeneous as well as heterogeneous catalysts. The methods that can be employed are photocatalytic or light driven, and people use catalysts, especially metals and metal oxides, as electrocatalytic medium. And we can estimate or evaluate the proton evolving catalyst by the same turnover frequencies for homogeneous catalysts and over potentials or Faraday efficiency for electrocatalysts. So these are some of the ways in which people try to measure the activity of a catalyst, and we have done. So here's an example of the cobalt oxime catalyst. So we as organic chemists know that one of the cobalt oxime complex is one of the most simplest compound that one can prepare, and many people have prepared this cobalt oxime catalyst with various substituents such as maybe pyridine, 2-pyridines, chlorides. Try to stabilize it by using BF2. Okay, so some stabilized catalysts are used, and this is one of the best known catalysts in terms of hydrogen evolution, which has been studied by Dempsey as well as Richard Eisenberg. And we thought that, and we have also in the literature, there has always been a combination of this ruthenium polypyridyl complex as a photosensitizer with the catalyst, that is the cobalt complex. Most of them have connected that by axial connection, where such a linker has been connected to the cobalt to form a complex, and have stabilized that by BF2, and the hydrogen evolution activity has been tested. There are other examples where other photosensitizers such as porphyrins, metal porphyrins have been connected to this cobalt catalyst and hydrogen evolution has been measured. However, the hydrogen evolution was not that great, nor could it be catalyzed or nor could it be driven by light. And
Hello.
yes, is my screen lost or?
Yeah, screen lost and voice was also lost, but now it had been restarted.
Yeah, now it's restarted. Okay.
Okay, screen is visible now.
Screen is visible now?
Yeah.
Yeah. Yeah. Right. Is the screen visible now? Yeah. Yeah. So we have the synthesis. We have is. Am I audible?
Sir, both
Yes,
the
sir.
things are quite clear. Screen is visible.
Clear now.
you are
Okay.
audible.
Okay. Screen is. I'm also audible. Great. Okay. So here is a synthesis of the complex that I was saying here. When you have a ruthenium bipyridyl dichloride, and we have synthesized the oxime. Then the oxime can be converted into further converted into this kind of a complex. From several ways we can prepare it. We can prepare it from synthesizing the oxime. We can start from the dione. We can give rise to this complex, and then when this complex is connected with the cobalt, when this is you add a cobalt to that. Okay, when we add. Okay. Yeah. When we add the cobalt, it can give rise to this complex, which we call as a diode assembly, where you have a photosensitizer. We have two photosensitizers. And a cobalt complex at the center with coordinated water molecule. Then we have added BF3 to that. It becomes a very stable complex. The stability increases. So this is very highly stable in solution and highly also soluble in water, because as you can see here, the charge on the complex is around 6+. And this is, I mean, the anion that can be present can be either PF6 or triplet or whatever anion that you want, you can add it. So this is extremely soluble in water and very highly stable in aqueous solution. And so this becomes a very good diode assembly for light driven water proton reduction. And we have done the regular characterizations that we need to do, such as UV visible spectrum, mass spectrum, the mass spectrum, and we have actually done the mass spectrum. We have seen the mass spectrum, and the peaks match to all the initially to the precursor, and then finally to the final complex with the cobalt in. And as one of the most important, as you can see here is the mass spectrum, where we can actually see that the calculated of the fragment and the observed values for several fragments match very well. One of the interesting aspects is that though rutheniums are diamagnetic, cobalt is a paramagnetic center at the cobalt 2 is a paramagnetic center, and we can show by doing an EPR that we get the cobalt that is coordinated here, because there is a question that is asked by the referee: how do you know that the cobalt is present in the system? Because cobalt is paramagnetic. So we actually recorded the EPR spectrum and of the complex, done the simulation and shown that cobalt is actually present over there, because the EPR gives rise to a signal for an unpaired electron. So we can, and other techniques such as cobalt ruthenium electrochemistry was done for the complex, and we can identify, like by differential pulse as well as by normal cyclic voltammetry, we could identify the ruthenium and cobalt species that are present, and as well as the ligand base that were also present in the system. And we have done several electrochemical techniques which will prove that the complex is quite stable. We have also done some single photon counting measurements for identifying that the complex is stable and can be utilized. And we have done transient absorption spectroscopy. I won't go into the detail, but when we shine the light on this complex, we can actually find out that it forms hydrogen. We have measured it by GC, and we saw that the light-driven hydrogen evolution, as you can see over here, we found out the turnover numbers, the turnover frequencies for this complex to be about 170. For the axial complexes, as you see, the numbers are quite low, the turnover numbers, with irradiation times which are around 4 to 8 hours in this case. But in our case, when we take our complex, we see that the turnover number is quite high, 170, at a very less irradiation time of 3.5 hours. So the diode assembly also based on some measurements, we have to assign the mechanism, which can also be proved by using various other techniques. But we have tried to plot a mechanism that there is oxidation of the ruthenium 2, and then followed by oxidation of the cobalt and sequential reduction to cobalt 2 species to actually generate hydrogen from the H plus that is given here. So you can, with the conversion of ascorbate, which is used as a sacrificial oxidant in this case, to give rise to hydrogen. So a mechanism has also been in place, a plausible mechanism, not a final mechanism, because some of the intermediates are not been identified, but a reasonably good mechanism can be put in place. So both the processes, one that is oxidation by ruthenium polypyridyl complexes, as well as reduction by a ruthenium cobalt ruthenium assembly, has been attempted. Full characterization of complexes has been done, and it has been shown that the turnover numbers and turnover frequencies for both the water oxidation catalyst as well as the proton reduction catalyst have been found to be better than the previous or previously reported complexes. So to summarize, amongst the present complexes, as I said, highest turnover number has been found out for the for the complex which has the glycol-uryl group. Maybe which is good because it forms a structure which helps in to attract and form proper channels for incoming molecules. We have also assembled a diode system wherein a ruthenium cobalt ruthenium, which has a high stability but a reasonably good moderate activity with excellent photophysical properties as well as electrochemical properties have been studied, and it will help us to develop better diode assemblies to be used in future photoelectrochemical cells. So this is so I would like to, in addition to that, we have also other complexes, because we have the assembly and the expertise to measure this complexes, we have done some collaborative work with people from ICER and other systems which have unusual structures and possibility of coordination of water as well as solubility in water. We have studied some cobalt complexes, some nickel complexes, and their activity has been studied. We have also studied electrocatalyst, but I will not go because that is more into heterogeneous. And we are, as I had presented here, here are some of the systems that we are still trying to publish. I would like to take this opportunity to thank our central instrumentation facility, where most of the characterization was done. And in addition to that, CEF, IIT Mumbai, CRI Lucknow, ICER Pune, NCL Pune for some of the collaborations. And I have several collaborators, not for this work, but other work. We do a lot of collaborations with our Bhumi Shankar at ICER, Eva Mari at University. And funding was provided to the student by SPPU, and CSIR New Delhi provided us some contingency, whereas some funding was also available to present this work by my student in some of the conferences in Japan as well as in Europe. Thank you very much for your attendance. I'll be happy to answer any questions that you may have.
I request participants to post their questions in chat box so that we can ask Professor Kumbar.
If anyone has any, please put in the chat box.
Yes, please.
No question. Now I request Dr. to express his views. Dr. Bhandi, sir.
There is no question in the chat box, so I think we'll conclude.
Yes, sir.
Professor Kumbar, I'm really thankful for your wonderful work, great work, and published in a very top-rated journals. I'm really pleased to see your results. They are giving very good high turnover numbers for hydrogen generation, and it's really great to see your talk. It's my pleasure that you are here and give excellent invited talk. Thank you very much. On behalf of organizing committee, I'm thankful to you.
Thank you. Thank you very much, sir. It's a pleasure to be always associated with with LVH. Yes, we have been associated with several aspects, and also to meet several chemists, especially inorganic chemists like Bhande sir and Kapadnis sir, and also to work together on
Thank you, sir.
Thank you.
Giving your valuable guidance to us, and your research topic was splitting of water by transition metal, means uranium and cobalt. Thank you, sir. Thank you, Bhandi, also. Now I request Iris sir to propose vote of thanks.
Thank you, madam. Good afternoon, everyone. I am here to present vote of thanks. On behalf of the entire organizing committee, I would like to express my gratitude to resource person, Professor Dr. Avinash Kumbar sir, for giving your valuable insights on the topic of water splitting by transition metal polypyridyl complexes, and chairperson, Professor Balchandra Bange sir, for chairing the session. Thank you for sparing your valuable time and sharing your immense knowledge with us. I would like to thank Principal Dr. B.S. Jagdale sir, Head of the Department of Chemistry, Professor Dr. K.H. Kapadnis sir, Professor Dr. T.B. Pawar sir, and the entire organizing team for giving me an opportunity to present vote of thanks. I would like to thank participants for your time, attention, and patience. I hope all of you enjoyed the session. Thank you all once again. Thank you, madam. Over to you.
Beyond the protocol, I would like to thank both my friends, Avinash ji and Balchandra ji, for accepting our invitation and delivering the lecture and share the session. Thank you very much. Thank you.
Thank you.
Okay, sir.
Thank you.
Thank you, sir. Now the third technical session of Dr. Chaudhary. Now the session was over to Dr. Chobe, sir.
Yes, ma'am. I am audible.
Yes.
So, welcome back again for the third session of this international conference organized by the Mahatma Gandhi Vidya Mandir, the Venkatrahire Arts, Science and Commerce College, Nashik. So, very good afternoon, one and all. I, Dr. Santosh Chube, feel extremely honored and privileged that this year our college is celebrating the golden years. And for reasons, here we are organizing an international conference on.
Professor, 1 minute. Professor Chaudhary is there in the attendees.
Yes, ma'am. Can I, sir?
Sir, you proceed. I have promoted into the panel.
So, sir, you continue. You continue. Avinaji, if it is possible, please mute your mic.
Yes, please. So, I, Dr. Santosh Chube, feel extremely honored and privileged that this year our college is celebrating a golden jubilee year, and for that, this session is being with us with the young scientist Dr. Moreshwar Chaudhary, who is fellow ICQ Spain. We are having a great opportunity to listen to the few words of this renowned scientist, Dr. Moreshwar Chaudhary, Spain. So I'd like to request Professor Dr. K.H. Kapani, sir, Head Department of Chemistry, Chairperson, BUS Chemistry, MGV Trust. Dr. Kapani, sir, please chair these sessions. Dr. Kapani, sir, valuable guidance. Many students is awarded PhD and more number of publication in his credit. So I would like to request Dr. Kapani, sir, please chair the sessions. And now is requested Dr. Vishnodra, sir, please introduce the young scientist, Dr. Moreshwar Chaudhary. Over to you, sir.
Let me take this opportunity. The responsibility had been given by the organizing committee to me to chair this session. I'm accepting your invitation. Thank you very much.
Thank you.
Thank you, Chobe, sir. Indeed, it's my great privilege to introduce the eminent speaker of this third session of international conference. For this session, we have eminent speaker Dr. Moreshwar Chaudhary with us. Dr. Moreshwar Chaudhary is expertise in continuous flow chemistry, polymer supported asymmetric organocatalysis, metal catalysis, that is homogeneous and heterogeneous, and also in synthetic organic chemistry. Dr. Moreshwar Chaudhary is currently a Marie Curie postdoctoral fellow at ICIQ Spain under the advisor Professor Michael Pericles. Dr. Moreshwar Chaudhary has also served as postdoctoral research associate at the IISER Pune from January 2022 to June 22 under the guidance of Professor B. Ganaprakasham at the Indian Institute of Science and Education Research, Pune. Dr. Moreshwar Chaudhary completed his PhD at the IISER Pune under the able guidance of Professor B. Ganaprakasham. More importantly, I would like to add here that the Dr. Moreshwar Chaudhary has published so many good reputed publications in the Journal of High Repute, such as Journal of Organic Chemistry, then Organic Letters, Asian Journal of Chemistry, then Journal of Organic Chemistry, Organic Biomolecular Chemistry, Organic Process Research Development, ACS Sustainable Chemistry Engineering, ACS Omega, and also he has published a book chapter entitled Metal Catalyzed Synthesis of Peroxide in the Theme publication. I would like to add here, he has received both Best Poster Award at the 8th International Conference on Frontiers in Organic Synthesis Technology, which was conducted in Hungary. He is awarded Marie Curie Doctoral Individual Fellowship for 2020 at ICIQ Spain. He has won Best Poster Award at the 4th UCAM MS Conference on Green and Sustainable Chemistry, Spain. He has also won Best Poster Award at Alexander I Kashid, India. DST served international travel grant for traveling to Spain for the research presentation. He has qualified that national level CSR UGC national eligibility test, which is conducted by CSR. So there are more number of publications and credit to under the credit of Dr. Morishwar Chaudhary. Sir, we are very fortunate to have with you with us the eminent personality, Dr. Morishwar Chaudhary, and definitely all our participants are going to benefit by your valuable guidance. Thank you, sir, for accepting our invitation for this international conference. Now I would like to introduce the chairperson of this session, Professor Dr. Kapadnis, sir. Dr. Kapadnis, sir, is working as a professor at LVH Art Science and Commerce College, Nasik. Sir is chairman of BOS in Chemistry, MJV Nasik. Sir is also ex-BOS member in Chemistry, SPPU Pune. I would like to point out here that more than 100 research articles are published in the journals of high repute. 3 students have been awarded PhD degree under his able guidance. 1 Indian patent is filed. Sir is member of editor advisory board of Indian Journal of Research Studies in Pure and Applied Sciences. Sir has received Best Teacher Award by Panchavati Sarvajanik Vachanalaya. Sir has received National Level Best Teacher Award. Sir has received Samta Fellowship Award. More than 84 articles are published with an impact factor greater than 1. 41 research articles have been published with an impact factor of more than 2. And 23 research articles have been published with an impact factor of more than 5. Sir has chaired a session in a 2 international and 1 national conference, presented number of research articles in the national and international conferences. Sir, it is great pleasure to have you with us as a chairperson for this session. Thank you, sir. Over to Chobe, sir.
Thank you, Vishnu, sir. So the session is open. Dr. Morishwar Chaudhary, please continue.
Okay. Am I audible?
Yes, sir. You are quite audible.
Okay, thank you. So I will just share my screen. Okay, my screen is visible now.
It is visible.
I will just move. Okay, so first of all, I would like to thank the organizing committee for this year CRCNS-2022 and Professor Vishnu Adole for introducing myself and all the coordinators, organizers of this event. And thank you for giving me this opportunity. The title of my talk for today will be Polymer Supported Asymmetric Organocatalysis in Continuous Flow, which will be a powerful strategy. At the end, I will convince you with this, whatever the process we develop, and it will be more of a brief introduction about the continuous flow processes as well as organocatalysis. So this is the place where I work, ICIQ, which is in the top 10 department of chemistry in worldwide. It has 17 research group with more than 200 PhD students as well as more than 70 postdoc students. So there are always exciting opportunities at ICIQ for PhD, postdoc, and master students. So the PhD funding was awarded by different funding agencies such as Marie Curie, Euraccess, and other European Commission agencies. As well as there are master program in ICIQ, as well as a neighboring institute called the URV. So this will be beneficial for the students who want to pursue their career in chemistry as a PhD student or a master student. So the city which I live, which is the Tarragona, it is in a UNESCO World Heritage, has a tourist attraction for beach resort, and it's very famous for olives and sauces. And there are the cultural similarity where the festival are celebrated called a human tower or castellars. So which is quite similar with the Indian festival, which we celebrate in India called the Handi. So coming to the other gate attractions of the city, which has this famous festival as well as the Roman architectures as well as amphitheater where the bullfights occurs. So now I will turn to the chemistry point. So the term chemical reactor appears for the first time in 1945, although the chemical reactions they proceed via the chemical reactions scientists are performing from 100 and 1,000 years before. However, the industrial revolution brought a evolution of chemical reactor also by putting a lot of innovation and money for making these sophisticated reactors. Now, if you see the history, I will just take the highlighter. So okay. So there was a reaction which we used to do in the open pot. Then it converted into the distillation apparatus with the helmet, and later it converted into the more sophisticated tubular reactors or a continuous stirred tank reactors. So as day passes, the size of the chemical reactor is shrinking nowadays, and one can see this very small chip reactor, which is a size of maybe 2 or 3 inches, can be used as a tool for performing the reaction in a continuous flow. So what is the continuous flow? Continuous flow is a process where the reaction performed continuously by pumping one or more starting materials. However, the instrument setup typically includes the different parts such as pumps, which used to introduce the reagents, then the reaction loops, then we have the TPs for mixing the two reactants. Then there is a coiled reactor, which is used to perform a reactor in a temperature control manner. For heterogeneous catalysis, the packed bed reactor is used, and the back pressure regulator is used to control the overall pressure, as well as one can do the superheating of the solvents in the given reaction. So there are 4 main types of continuous flow systems. Type 1 consists of the pure homogeneous one, where the reagents are in homogeneous phase passed to the reactor to get the product. Second type is the use of supported reagent, which is packed in the packed bed column, and reagent A is flown and reacted with the supported reagent B to get the product. Third type is the homogeneous catalytic system, where the reagents and catalyst is in the same phase, and they reacted in a reactor to get a product. However, the last catalytic system is the supported catalyst, where any heterogeneous catalyst is packed in a glass bed, and reactants are flown over it to get the desired reaction. What are the different types of reactors? This is the very small reactor called a microchip, and this microfluidic device is used for the reactions of proteins, sugars, and very small scale chemistry. Then there is other type of reactor called a tubular reactor. This is nothing but a simple PTFE tube which acts as a reactor. And then there is the last one is the fixed bed reactor, which is used for the heterogeneous reaction, which can be made up of glass and PTFE. So how a continuous flow setup look like? This is on the left hand side, the commercial reactor I have shown, which is from Vaportech, where you can see the HPLC pumps are used to deliver a liquid into the glass coated reactor or for a heterogeneous catalysis. So the cost of the instrument is little high, but if you see the customizable continuous flow setup, where we can see the normal syringe pump is used to deliver reagents A and B from both the pumps, and then the reaction will occur in the PTFE tube. For heating or cooling purpose, simple oil bath or water bath also can be used if one want to customize the less expensive reactor. So why flow chemistry? If you see the principles of green chemistry, most of the principles are fulfilled by the continuous flow chemistry, such as the reactions in continuous flow are rapid and they give byproduct minimization as well as a safer synthesis. The very important factor in continuous flow chemistry is the greater surface to mass ratio as compared to the simple batch chemistry. Next advantages in continuous flow is superheating of solvent. So what is superheating? Superheating is nothing but heating a solvent above its boiling point. Let's say DCM has a boiling point of 39 or 40 degrees Celsius, but by using continuous flow setup and by placing some back pressure regulators, we can actually heat the DCM up to 100 degrees Celsius. Then another advantage is the size of these flow reactors are very compact, so less space is required, and real-time analysis is also possible. However, there are some disadvantages, such as special types of reactors or the pumps are used for the reaction to carry out with the solids, and the cost of the instrument is also little high. But if one customizes the reactor by using homemade or from local vendors, so it will be a cheap option. So with this introduction, now these are the applications which we have shown using a continuous flow setup at IISER Pune with the rearrangement of peroxide, as well as we perform the peroxidation. We have done more work in a continuous flow, such as use of magnetic nanoparticles for continuous flow reactions using a heterogeneous medium. So with this, now I will move towards the introduction of asymmetric catalysis. So over the million of years, there is a continuous evolution where nature has perfected its machinery and it performed the chemical reaction in a highly enantiomer manner. So till today also, chemists cannot mimic the nature in 100% way. So basically, chemists uses 3 strategies to mimic the nature. First one is the metal catalysis, then the biocatalysis and organocatalysis. So in today's talk, I will be more focused on the organocatalysis. So what is asymmetric organocatalysis? So nature is full of objects that are chiral. Let us take example of our hands that exist in a pair and which are a non-superimposable mirror of each other and which cannot be superimposed. So the asymmetric catalysis is organocatalysis is nothing but a method for preparing a chemical compound by some biased way in which synthesis of producing one stereoisomer over other using an organic molecule. So why asymmetric synthesis? So if we see the example of ephedrine, there are 4 stereoisomers where 2 of them are biologically active, however 2 shows some toxic activity. So for this reason, different enantiomers have different biological activity. That's why it is very important to synthesize the specific enantiomer to avoid the unwanted side effect of another enantiomer. However, if we particularly synthesize one enantiomer compound, then the amount of dose needed to deliver into the patient is required very less. So what are the criteria for ideal organocatalyst should be? It should be stable for a long period of time. It should be stable under moisture and air. It should be highly selective and it should induce chirality as well as high enantioselectivity. It should be easy to handle as well as inexpensive. There are so many conditions for a catalyst to be an ideal catalyst. However, in my opinion, ideal catalyst should be infinite and it should never die after performing many reactions. So if we see the past 2 decades, there is a steady increase in the number of publications in organocatalysis, and this trend will continue to grow on. The asymmetric organocatalysis basically of 2 types. First one is the covalent organocatalysis, and another one is a non-covalent organocatalysis, which proceed via hydrogen bonding or a counter anion directed synthesis. So there are 5 actuation modes of chiral induction. First one is the which involves the enamine catalysis, then the hydrogen bonding catalysis, iminium catalysis, SOMO catalysis, and counter anion directed catalysis. The enamine catalysis, which was first demonstrated by Hakos and Parrish in 1971. This one was the first example to use proline catalyst for the asymmetric reaction. However, later Lees, Lerner, and Barbas, they developed the chemistry of the proline catalyst. In India also, Professor Kumaraswamy in 2008, he also demonstrated some examples using proline as a catalyst. Then in hydrogen bonding catalysis, use of thiourea has been done, and the reaction proceeded via activation of LUMO, and which was demonstrated by Jacobson and E.J. Corey. Later in 2000, by using a iminium catalysis, Macmillan showed the use of Macmillan's catalyst for performing the asymmetric reactions. However, he also introduced the SOMO activation, where single electron transfer can occur in the catalyst in presence of adding sacrificial reagents. And later, the counter anion directed synthesis also was developed by Jacobson in 2007. So, if you compare the homogeneous as well as heterogeneous catalysis, both of them have their advantages and disadvantages. So, I'm just showing a little flavor of what are the advantages we associated with each other. And this is not I'm not comparing these 2 things because they have their own merits. In homogeneous catalysis, they have high diffusion, greater selectivity. Fine tuning for in-situ selectivity is easy, but they are expensive. Separation problem at the end of reaction. Heterogeneous catalysts, they are easy to purify, recover, and reuse. They are more thermal stable. But fine tuning of heterogeneous catalyst is sometimes difficult. There is a mass diffusion problem also occurs, and catalyst deactivation also possible in most of the case. If you see the industrial catalytic process, all processes in the industry, out of that 85% are comprises of catalytic and 15% are non-catalytic. In catalytic processes, again, 80% are heterogeneous ones and 17% are homogeneous one. However, the minor one was done by a biocatalysis. The typical example of heterogeneous catalysis is Haber-Bosch ammonia synthesis as well as the Ziegler-Natta polymerization. So why we need to support the organocatalyst? If we see the cost of the commonly used organocatalysis, such as the TRIP catalyst developed by Benjamin Lees, as well as the Macmillan catalyst developed by Macmillan, and for their great discovery, they have been awarded with the Nobel Prize. Again, we see the structure of Jorgensen IC catalyst, which is air sensitive, and price is again little high. So for that reason, we need to think alternative way how we can minimize their use and continuously reuse such type of catalyst. So why need of supported organocatalysis? The idea is transforming homogeneous catalytic process into the heterogeneous one, owing to their advantages such as easy purification, as well as more robustness and thermal stability, and they can be used in a continuous flow applications where inline recycling with a linear increase in turnover number as well as turnover frequencies, and it is possible to perform a site isolation by a volt lamp scenario, meaning that both the active sites such as acidic as well as basic can be incorporated in the same catalyst. So what was the inspiration where it started, the solid polymer supported catalysis? In around 1970, 1975, Robert Merfield has pioneered the use of polystyrene resin for the solid phase peptide synthesis. So for his awesome work, he was awarded with the Nobel Prize in 1984. What he basically did, he took the polymer, incorporated some suitable group which will be lived easily, and he reacted the amino acid with the polymer supported resin to get the amide. Then he reacted with the another amino acid by using a sequential protection deprotection strategy, and by using this approach n-fold repetition of amino acids can be occur in a very simple way, and at the end the resin can be cleaved by using a selective deprotection. So when not to support your catalyst, first we need to carefully check the chemical compatibility of the support. Chemical compatibility in terms of decomposition as well as solvent capability. There is possibility of very slow reaction in heterogeneous catalyst in some cases, and there is also possibility of loss of chirality during a modification. This example I will share in my next slides. And later, if we don't have an element than carbon and hydrogen, then there will be a problem for characterization, because heterogeneous organocatalysis has a drawback, one more drawback, which is the limited characterization techniques available for this transformation. So how to design a supported catalyst? Basically, it has three main components. First one is the catalytic site, then second one is the linker, and then the polymer support. Here in typical example, I have shown the increase of steric bulk for inertial control, then placing of spacer to keep the polymer support away from the catalytic site. So if we fulfill these three criteria, then it is possible to get a supported catalyst which will show a high activity in chemical reactions. So what are the different type of supported materials for supporting the catalyst? First one is the polystyrene, and we most commonly use this support in our laboratory for performing the heterogenization of the homogeneous catalyst. Second one is the magnetic nanoparticles, where use of iron, cobalt, and other species can be used to anchor active site onto the nanoparticles, and at the end of the reaction, it can be separated by using a simple decantation technique. Next, the zeolites also can be used as a support by a chemical modification techniques and can be used for the supported catalysis. Another one is the use of a silica. It's also explored in many reactions. Use of ionic liquids are also demonstrated by various research groups over the past decades. And interestingly, use of organotextile catalysis also developed by scientists for performing the reaction in a heterogeneous manner. So what are the types of polystyrene resins are there? Very important and most commonly used Merrifield resin, which is available commercially or can be prepared in lab by using the simple polymerization technique by using a readily available surfactant as well as radical initiators. Another resin is the Wang resin, which these two resins showed the good swelling in non-protic solvents. However, the other resins such as Janja gel and Tenta gel, which shows the good swelling in more polar solvents. So by using a different strategy, trial and error methods, by balancing the molar ratio of cross-linker as well as the polymer as a styrene as well as divinyl benzyl, Perica's group has pioneered the field in polymer supported organocatalysis. These are the catalysts I shown which were developed by Perica's group at ICIQ and University of Barcelona over the 2-3 decades. It's ranging from the polymer supported prolines as well as polymer supported Macmillan catalyst, Jorgensen-Hayes catalyst. Then we have polymer supported List catalyst, which was developed by Benjamin List. Then we have the polymer supported Jacobson thiourea catalyst. And we have the synchronoalkaloids, which supported on the polystyrene. And then we have some amine-based catalysts also. And there are the catalysts which was supported using a metal also, which I not shown here. So out of this, basically I work on polymer supported TRIP and polymer supported Jorgensen-Hayes catalyst, as well as I have also involved in the synthesis of few novel immobilized catalysts, which I not shown here. So according to the Sir John Conforth, ideal chemical process is that which one arm operator can perform by pouring the reactant into a bathtub and collecting the pure product from drain hole. This statement was given by John Conforth, a great enzyme catalyst chemist, where he was awarded with the Nobel Prize in 1975. So by merging the supported catalysis in a continuous flow manner, which will lead the reaction to the ideal chemical process. So with this introduction, now I will show how we synthesize the different polymer supported catalysts. Synthesis was started with the simple S-binol, followed by protection and bromination, followed by Kumada coupling. Then we deprotected the MOM group to get the binol derivative, which one again with the bromination afforded the dibromo species, which again with the reaction with the styrene derivative using a Suzuki coupling, we got the monomer. Now this is the crucial step where we can perform the cross-linking polymerization in presence of polyvinyl alcohol as a surfactant, and then AIBN as a radical initiator. And styrene, and we have used a styrene as well as boric acid in this transformation to boost the reaction. So after the simple polymerization step, here one more thing we need to consider, that by keeping the parameters to balance the stoichiometry between styrene, 1,4-divinyl benzyl, and monomer. Fixing this stoichiometry is little bit tricky, and if this stoichiometry goes wrong, whole polymerization step will be very difficult to get a good catalyst. So the polymer supported TRIP catalyst is air and moisture stable and can be stored for years, because such type of catalyst was developed in 2017 also. So, and the characterization for this catalyst can be done by using elemental analysis of percentage of phosphorus present in the polymer, and we can get the actual functionalization and molecular weight. So what is the functionalization here? Functionalization is nothing but, in this case, for example, 0.1356 millimole per gram. It means 0.1356 millimole of active catalyst present in 1 gram of overall polymer. So after making this catalyst, we tested its activity for the model reaction in a batch as well as continuous flow. Both gave the similar enhanced selectivity and decent yield. However, after the reaction, it's very easy to reactivate the catalyst by simply washing it with a HCl solution in ethyl acetate. Later, the main objective using this catalyst was to perform the cyclization reaction. So this is the typical setup where we have used the syringe pump to deliver the liquid or a reactant, and we can here I loaded the polymer supported catalyst, and here this is the BPR and this is the collector to get the reaction mixture. Along with the polymer supported trip, we check the spinol as well as polymer supported thiourea catalyst also for this cyclization step. After extensive optimization and many reaction conditions, which I not showed here, but I just show the main results where we got the maximum enantiomer selectivity of 68%. However, the conversion was very low for this transformation, and for that reason, we thought to develop a new catalyst, which will be comprises of camphor sulfonic acid. So we started to synthesize the polymer supported camphor sulfonic acid. And synthesis started with the very simple commercially available aldehyde by deprotection, followed by reduction and then in hankering of the very filled resin onto the alkenyl benzyl alcohol afforded the resin. The reaction of this resin, which was done with the 7-azido camphor in presence of DMF and copper iodide via click fashion to get this resin. So preparation of this 7-azido camphor is where it was very difficult part, and it involved a series of Wagner-Mervin rearrangement, and I started with very large scale, but I able to get very small amount of the compound for this reaction. However, at the last step was to introduce a sulfonyl group here, which will act as a acid for the cyclization step, but it turned out to be that in last step, the enantiomer pure part of this camphor unit was completely destroying in sulfonation step, and we finally ended up with the sulfonation over the aromatic parts, and the catalyst was racemic, and we couldn't able to proceed the next reaction using this catalyst. So we are still developing some other catalysts for this transformation, and we will hopefully get through the good enantiomeric exit as well as yield for this transformation. So in other project where the use of Jorgensen Hayashi catalyst was done for the another type of asymmetric Michael addition reactions. For that reason, we synthesize a library of Jorgensen Hayashi catalyst. The reason was, the idea was to incorporate the CF3 group on the aromatic part, and it may lead to give the more robustness as reported in the literature, but it turned out to be something different, which I will share in my next slide. So why proline? Because it has versatile tunable groups. It's very small molecule and give a nice hydrogen bonding. Here we incorporated the TBS as a protecting group and incorporation of aromatic part, which will give the steric control for this overall reaction. So the synthesis started with the simply commercially available 4-hydroxyproline in presence of acetic anhydride with this lactone, which broken in presence of acid and again protected with the methyl chloroformate to give this species, where the Grignard addition across this ester afforded dihydroxy species, which on reaction with the 4-chlorostyrene afforded this oxalizidone ring, where it's broken in presence of KOH ethanol and protected with the TBS triplet. After getting the monomer in hand, again we tried to do the copolymerization technique using a emulsion polymerization, where the reaction occurred in a 2-phase. Water was one phase and toluene was the other phase, and use of surfactant to attract the hydrophobic part was and to generate the micelle formation was the driving force for this reaction. So this is well known in literature. So how one can do the emulsion polymerization, but in this case the optimal stoichiometry between the styrene 1,4-divinyl benzene and monomer also was very important. So this one was the key point for this polymerization step. So after performing the polymerization, we could able to get a very nice solid, which is gel in polar solvents. The prepared polymer was characterized by using elemental analysis, and it shows the functionalization of 0.7357 millimole per gram. However, we tried to characterize it by using a 13C NMR in a cryoprobe mode, and we found that this exactly matches with the monomer 13C peaks of the monomer which we use for this transformation. So this is the swelling test performed of the supported catalyst. You see, in absence of solvent, the catalyst is a nice white solid. As soon as we add a DCM into it, it will show a very nice swelling. So the key point is here, the incorporation of the active sites are inside the pore of the polymer. So the reaction are happening inside of the pores of this polymer. So if there is no swelling in the catalyst, this overall reaction won't proceed. So therefore, we need a swelling for the catalyst to reaction occur. Another way to synthesize the supported Jorgensen IST catalyst is a click approach, where we started the reaction with the Grignard adduct and reacted with the propargyl bromide to get this O-alkenylated species, and the protection of this the carbamide group, we got this hydroxy compound which protected using TBS triplet to get the alkyne monomer. Here, the azidopolystyrene was prepared from berry-filled resin. Simply, you need to take the berry-filled resin and react it with the sodium azide to give this azidopolystyrene compound. So now the key step is the click reaction between this azide polymer and alkyne in presence of copper catalyst. Straight away will give the triazole linked polymer supported Jorgensen IST catalyst. So the reaction monitoring was performed by using IR, and at when the peaks of alkyne was disappeared and the peaks of azide disappeared, we concluded that the reaction proceed through a nice way and we can stop the reaction. Later, by elemental analysis, we found that functionalization was 0.4071 millimole per gram. And this catalyst is also air and moisture stable and can be stored for many months and years. As compared to the homogeneous catalyst, which is air stable, this catalyst is highly stable. The main thing is with the berry-filled resin is the shape of the polymer is very nice, spherical and evenly distributed. However, in the homemade cross polymerization technique, the shape of the catalyst can be disturbed. After synthesizing the catalyst, we thought to use it for isometric Michael reaction. So this asymmetric Michael adduct can be served as a precursor in varieties of transformation. So for that reason, first we tried to perform the reaction in batch, and we tried to check the activity in presence of additives. Interestingly, without CF3 group, simple Hayashi catalyst, it gives the reaction well in presence of acetic acid. However, the Jorgensen catalyst, which has CF3 group, which work well with the calcium triplet. If we add acetic acid along with the Jorgensen catalyst, this is not giving good yield. So the reason for this is still unknown, and we are still trying to find it. Why this like the selective behavior is happening. So the solvent DCM was the best solvent, because in other solvent enantiomeric excess was drastically dropped, and the reaction time was ranging from 24 hours for Jorgensen catalyst and 36 hours for Hayashi catalyst. After that, we perform a substrate scope with the newly prepared Jorgensen catalyst, and the same catalyst is used for making the different substrate. The yield was ranging from good to moderate to very good for the substrate, and electron donating groups, they give very poor yield. Later we tried to do optimization for continuous flow process using polymer supported catalyst. The typical setup involves the packing of polymer supported catalyst in omni-fed glass reactor and flowing the liquids using syringe pump or HPLC pump, and at the end we can get the reaction mixture. The process was monitored by using a NMR conversion, and enantiomer selectivity was determined by using a chiral HPLC. Here, it's important to show that reaction with the Jorgensen catalyst at very low flow rate in place of calcium triplet as additive gave 94% EE, and residence time was very long for this transformation. However, using a Hayashi catalyst, we could able to get 96% EE in 47 minutes of residence time. So here, what is residence time? Residence time is nothing but how much time of the reactants are spending in the reactor, which is known as the residence time. So after these results, we found that the increasing the concentration is giving rise to more yield. So for that reason, we thought to check the reaction in absence of any solvent. So what we did, we took a highly concentrated 50 millimole of cinnamaldehyde along with the dimethyl malonate and acetic acid. We mix them in one vial and flow in this solution to the packed bed of Hayashi catalyst, which is around 1 gram. 1 gram catalyst we loaded into this cartridge, which has approximate size of 3 to 3.5 centimeter. And then we set the temperature using the outer jacket, so at 60 degrees Celsius, and we put a back pressure regulator with a 75 psi. So the flow rate was set to be the 0.05 per minute, and at the end of the reaction, we could able to get 5.5 gram enantiomer product in 4.33 hours. So the residence time for this was 55 minutes, and turnover frequency is 1.21 gram per hour. So simply washing this catalyst with acetic acid, it can be reactivated and it's ready for next reaction. So similarly, this enantiomer Michael adduct, which obtained in this transformation, is useful as a precursor for phenoxetine. However, we perform the next reaction by using a 4-fluorocinnamaldehyde in a similar fashion, and we could able to get a 5.62 gram product in 4.33 hours. This reaction is also solvent free, and it has turnover frequency of 1.3 gram per hour. So if we project the synthesis, 1 gram of catalyst, it may give 25 gram in 24 hours. So the idea is to put 10 columns of 1 gram each to get the 240 grams of product in 24 hours, or one catalytic system with a 10 gram catalyst will give 240 gram of compound in 24 hours. So this will serve as a micropilot plant for high process intensification. So industry always look for such catalysts and such processes for making the bulk scale production. It's very important to mention that we use same catalyst for optimization more than 150 reactions, and the catalyst is still active and it is immortal. So later we try to focus on the long-term monitoring of this reaction, and then we check the reaction progress by using 1H NMR, and enantiomer selectivity was monitored by using HPLC. It is important to explain that from the initial point to the end point, although the conversion drops slightly, but enantiomer selectivity is not dropped. It's showing the robustness of the catalyst. So with this example, now I will show the later part of my the same project, where we use the Michael adduct for synthesis of indole-quinolizidine in flow. If we see the structure of roimbine, reserpine, histidine, the common thing is that they have the typical stereochemistry at the C12 position, where depolymerization step is crucial. And these natural alkaloids have shows the very potent biological activities. So this indole-quinolizidine derivative is non-natural derivative, and By looking at it, first thought come in our mind is its complex structure. So that's why, due to the complexity, they are not much explored for the biological activity. So if we do the retrosynthetic analysis, everything looks very easy, where we selectively break these bonds and we can get our starting materials. So what we achieved, we got a single diastereomer in flow the first time, and then the residence time was drastically reduced from 16 to 20 hours to 50 minutes. We can actually do the thermodynamic as well as kinetic control to get exactly the cis and a trans isomer by controlling the temperature. Both enantiomers are accessible as we have both the enantiomeric catalysts. So with this idea, we have proceeded for the optimization of this diastereoselective process, where we varied the concentration of TFA. The reaction is very dependent on the concentration of TFA, which required a 2.5 equivalent of TFA to carry out the to get the good yield. We also changed the flow rate. Higher flow rates was not giving the good yields. However, lowering the flow rate and increasing the residence time to 50 minutes, the reaction gave almost 90% EE with 76% isolated yield. After obtaining this optimized condition, we moved for the surface scope, and we performed the reaction of the different electron neutral, electron withdrawing groups, and we got the good to excellent enantiomeric excess and good yield for this transformation. The absolute cis configuration was assigned by using a single crystal exorography. So what are the future objectives for this now? So we are trying to do the telescoping of this continuous flow setup, where telescoping means one can do the reactions without isolating the product. One or two or more reactions also can be done in telescope manner. So idea is to get the Michael adduct in a continuous fashion and just introduce the TFA and tryptamine to get the endocrinologine derivative. So with this, I conclude my talk, and I gratefully acknowledge the organizing team CRCNS 2022 for giving me this opportunity, all the dignitary, chief guests, and all the scientists across the scientific community. I also acknowledge Professor Michele Pericas, my postdoctoral supervisor here at ICIQ Spain, and financial and research support from ICIQ, Marie Curie Actions Fellowship, and European Commission is gratefully acknowledged. And I would like to also acknowledge my family and friend for their constant support, and I'm very happy to take any question. Thank you so much for your kind attention.
Thank you, Dr. Mauricio, sir. Now I would like to request Professor Dr. Kapani, sir. Please, sir, over to you, sir.
Yes, thank you. Thank you very much, Morishwar Chaudhary, sir. A young scientist had been delivered his lecture quite satisfactorily. He had been covered all the aspects of the expected international conference presentation. May I now request the participants, if at all there is any question, please post it in a chat box. There are questions. No. Very informative session. Thank you very much. So please, if anybody is having any question, please put it in a question and answer. Till the time there is no question.
Sir, I would like to ask one query.
Definitely. Dr. Vishnu Adode, you are allowed to do so.
Sir, I would like to know the price of that flow chemistry reactor and what sizes of flow chemistry reactors are there, and can a laboratory like ours can afford it? What is your opinion, sir?
Okay, so the cost of the commercial Vaportic series, which I showed, which is around 25 lakhs. so it is quite expensive. But the option is to make it a customizable one, so which will be affordable, and we can buy the different components. There are main three components, which is one is to syringe pump to deliver liquids in a controlled flow, and then another aspect, another part is the PTFE tubing, which is also available from the local vendors in India. And the next part is we can do the homemade heating system, where we just need to put the oil bath or water bath, and we can heat the system or cool down the system. So for a very basic level, if we do the customization, it is very easy to establish a continuous flow setup in very low cost, as compared to if you go for the commercial one, which is quite expensive.
Okay. Is there any more question?
So there is one question I can read in chat box. How easy can microreactor technology be used for scaling up? So, so this is by Milin Pimpare. So there are two different things: microreactors and continuous flow reactors of different sizes. Let's say microreactor has very small volume size, which is around 150 microliter, which I showed in one of my slides. So if I go back, here somewhere it is. Okay, here. So this type of reactor, they have very small volume, up to 150 microliter. And if you want to scale up, so the parameters, once we optimize in flow, that will be same for scale up. Because let's say I optimize reaction using 150 microliter reactor, and if I want to scale up it, I need to use a similar series of reactors. Let's say I want to maximize into 10 times, then need to use the 10 reactors, or just need to increase the length of the overall reactor to get the desired product in more. We need to get the yeah, we can scale up those things quite easily. And in case of the other reactor, which are the simple flow reactor, where the size can vary from 5 ml, 2 ml to 10 ml and liters. So this depends what reaction you are doing. So microreactors basically used for proteins, then sugar, and very small scale chemistry.
Is there any more question from participants? Okay, if it is not there, we'll have to proceed. All the participants are requested to fill the feedback form. Feedback link had been posted in the chat box. Okay, let's come to the lectures. Sir had been started his presentation by explaining the evolution of chemical reactors. Then he explained different reactors like silicon-based reactor, tubular reactor, and fixed bed reactor. He focused on continuous flow reaction setup. Then he discussed the various advantages and disadvantages of flow chemistry. And after that, systematic organocatalysis. Sir then addressed why organocatalysts are important for symmetric synthesis. After this, Sir explained different types of supports for the development of supported organocatalysis. He also explained synthesis of polymer-supported S-strip catalyst. Then he presented the synthesis of library of Jorgensen Hayashi catalyst. The main focus point were development of active organocatalysts and the stability, NCO selectivity, and optimization of various parameters. At the end, he emphasized the synthesis of various systematic molecules using asymmetric organocatalysis under solvent-free conditions and optimized reaction conditions. Sir, your lecture was very informative and it was very good, self-explanatory. So thank you very much, sir, for delivering the lecture in this international conference. And thanks to the organization who have been given me this opportunity to chair this session of such a wonderful researcher. Thank you very much.
Thank you, sir. Dr. Arun, sir.
Yes, sir.
So can I come?
So let me propose the vote of thanks. I would like to add here one point that the 2021 Nobel Prize was awarded to the Benjamin List, who is also working in the field of asymmetric organic synthesis, that is catalyzed by asymmetric organocatalyst. And the same topic, under the same topic, Dr. Moreshwar Chaudhary is also working. So it is definitely a proud moment for all of us that you participated and delivered the talk on the asymmetric organic synthesis using asymmetric organocatalyst. Thank you, Dr. Moreshwar Chaudhary. Definitely, your talk was very wonderful, knowledgeable, and we all are immensely benefited by your talk. And in the future, our association will continue further and it will strengthen and we will develop more collaboration to conduct such a type of conferences. Thank you very much again for sparing your valuable time and sharing your immense knowledge with us. I would like to thank also Professor Dr. K.H. Kapadnis, sir, who chaired this session and guide us. Thank you once again. Thank you all participants for listening this wonderful talk. Over to Chobe, sir.
Thank you, sir. Thank you, Dr. Moreshwar Chaudhary, sir. You have to cover all the types of the reactor in very precise manner, and hope so all the participants can gather you share your views. Thanks a lot, sir.
Thank you so much. Thank you so much.
Advocate sir. Advocate sir. Inform about inform all the participants about tomorrow's program.
Yes, sir. So
schedule.